हॅलो हॅलो.
येस सर.
हा, गुड आफ्टरनून म्हणलं.
गुड आफ्टरनून.
ऑडिबल आहे. आमचे जॉईन झाले नाहीत का? हा, झाले. हजारे सर झाले. हजारे सर.
हा, गुड आफ्टरनून सर.
हा, गुड आफ्टरनून. कुलदीप आहेत का?
हो सर, हो आहे.
गुड आफ्टरनून ढाली सर.
हाय, गुड आफ्टरनून.
गुड आफ्टरनून ढाली सर.
गुड आफ्टरनून सर.
Good afternoon, sir. Raut sir, good afternoon.
Good afternoon. How are you?
I am fine, sir.
What to say?
I think voice is not coming.
Sir, actually, we had muted it. We are
Okay.
waiting for just 2 or 3 minutes.
Fine.
Yes, sir. Limse sir, namaskar. Dear participants, we are going to start in a short while. Just waiting few arrangements to be done. Please wait. Learn good. So, very good afternoon to one and all.
Afternoon.
Yes, sir. This is Dr. Deshpande, Assistant Professor and Head Department of Animal Nutrition and Co-Coordinator for this training program. I welcome all the guests as well as the participants joined for this program from the nook and corner of the country. And this training program, it will be almost for the information of participants, this will be almost a 10th program of this level from PGIVAS Akola. And before this program, we have also organized several international as well as national training programs on different topics. So this is a very important topic which we are going to deal for next 3 days, 2 days. And for today's function, this inaugural function, the chief guest is Dr. Arindan Dhali, who is a principal scientist in animal biotechnology in National Institute of Animal Nutrition and Animal Physiology, Adugodi, Bangalore. And the guest of honor will be Dr. Chandrasekhar Rao, Director, Interdisciplinary Research, Dr. D.Y. Patil Medical College, Hospital and Research Center at
Good
Pune.
afternoon, all.
Good afternoon, sir. So I wholeheartedly welcome all the guests, and the chairman of today's function is our beloved Associate Dean, Professor Dr. A.U. Bhikane, sir. So sir is also present here. So I welcome to you, sir, also. So first of all, I request Dr. Sunil Hazare, sir, who is the coordinator for this training program, to give a short introduction of how this program is going to be conducted for next 2 days. Dr. Hazare, please.
Good afternoon to one and all. Respected Chairman of inaugural function of this online training program on laboratory animals in biomedical research. Respected Chairman Dr. A.U. Bhikane, sir, Post Director and Associate Dean, PGIVAS Akola. Chief guest of today's function, Dr. Arindam Dhali, Principal Scientist, ICR, National Institute of Animal Nutrition and Physiology, Bangalore, and guest of honor who is present here, Dr. C.J. Raut, Director, Interdisciplinary Research, Dr. D.Y. Patil College, Medical College, Pune. In this online training program on laboratory animals in biomedical research, more than 190 participants have registered. I congratulate and welcome all participants joined here from different corners of the country. Most of the participants are students from life sciences and veterinary field. Also, participants from government sector, industry, and from research and academic institutions have also joined this 3 days training program. Indian pharmaceutical industry growing at a very rapid pace. It is a hub of drug and vaccine development of world. Laboratory animals are largely used by these industries. There is a high demand of technical manpower in the area of laboratory animal research in pharmaceutical industry. But there are no degree courses available in laboratory animal science in India. Some diploma courses are coming out nowadays. Hence, such training programs are utmost important to get good knowledge about ethics, rules, regulations in animal experiments, and the kind of experiments performed on laboratory animal in biomedical research. This training would also be very helpful to develop the interest among the students so that they can plan their research on laboratory animals, and they can see this field as a very good career opportunity too. In this 3-day training program, we have included very experienced academicians like Professor Dr. Pritam Mohan, Dr. Sole, Dr. Panchabai, Dr. Bankar, and noted scientists from renowned institutes like Dr. Vikas Dighe, Dr. C.G. Raut, Dr. Pravin Bankar, and Dr. Pranjali Pore. We have also included the lecture of industry person, Dr. Prahlad Bankar, who has established his own contract research organization and research laboratory at Pune. With these few words, I once again wholeheartedly congratulate and welcome all participants. Thank you.
Thank you, sir, for brief introduction of this training program. We are confident that during this training program, the participants will have a complete know-how of various aspects of use of these laboratory animals in life sciences research, and various aspects like breeding management, and other things, that how they are going to be used in research for carrying out different types of research. So that will be covered throughout this training. And I would request that participants would be making notes of whatever lectures they will be listening, as well as whenever they have any questions. So the participants, they are requested to write their questions in the chat box during lecture. If there are some questions, so that could be typed in the chat box. And after that, we will be giving chance for a question answer session for each speaker. So that will be going on very smoothly. Right now, I request today's chief guest, Dr. Arindam Dhali, to deliver his lecture as well as give his guidance to all the participants, and kindly start his lecture, sir. Thank you. Yes.
Thank you. Let me share the slides first.
Yes, sir.
Just a second. Yeah, is visible?
Yes, sir.
Okay, all right. Honorable Associate Dean and Course Director, Dr. Bhikane, Honorable Chief Guest, Dr. Chandrasekhar Raut, Course Director, Dr. Sunil Hazare, and other coordinators. Thank you for inviting me for the inaugural program of National Online Training on Laboratory Animals in Biomedical Research. Indeed, I am honored for the invitation. Now, let me express, say, over the last 20 years, we have received several notifications. Even I have personally attended, coordinated different type of training programs. But say a training program on laboratory animal research, its uses and other related issues are really unique. I have never seen such a training program. And I personally believe it is one aspect of the veterinary science which demands much attention. Now, almost 30 years back, when I joined BVSC, I remember our only exposure to laboratory animal was collecting blood from the ear vein, rabbit ear vein during pharmaceutical classes. And there is no formal courses on laboratory animal research or management. I'm not sure if it is there nowadays, but it is a very exciting and almost untouched area of the veterinary science. Now, if you go through the history of biomedical science, you will find remarkable achievements in this particular area is directly dependent or indirectly dependent on the usage of laboratory animals. So they are very, very crucial for the current and future biomedical research, because when you conduct an experiment on a laboratory animal, they give us the knowledge of the whole organisms, and which cannot be replicated in any way in any in vitro models, and that is very, very important. So typically when you talk about the laboratory animals, the first things comes in our mind, these are the rodents or rabbit. But as a matter of fact, the porcine, canine, feline, primates and ruminants are also included the laboratory animal for different experimental research. And this large ruminant and the primate, porcine, canine, feline, they are preferred model because because they are more complex organism, and in terms of their physiology, anatomy, and the body size, they mimic more closely the human and of course the veterinary applications. But there are problems and limitations using these animals. Very stringent regulations are there for using these kind of animals. And then these animals demands a huge space requirement. It's a very big setup you require to maintain such animals. And then moreover, maintaining them demands a high input cost. So here they are. So that's why laboratory animal research is very, very important. So there are several benefits. The first is that they have a short lifespan. You can complete a study in a very short period of time. And if situation demands, you can also do over the generation, which is very difficult with the large ruminant and primates or other related big animals. They also mimic the conditions required for the human and veterinary applications. And they are quite easy to maintain under controlled climatic conditions. They demand less space, there is less input cost, and there are also less regulatory restrictions. Typically, when you apply, say, clearance to your Institute Animal Ethics Committee, and if you are conducting research on rodents or rabbit, henceforth I'll refer for this particular lecture, laboratory animals as the mice, rodents, or rabbit, not the large animals. So typically, if you apply for the clearance, it is very easy because that comes under the purview of the Institute Animal Ethics Committee, and it will be clear itself. But if your experiment involves a cow or sheep or goat or other large animals, it will go to CPC ACA, and then there is a long process. So even regulatory restrictions are also less in case of laboratory animals. Now look at this figure. Over the past 107 years, 73 Nobel Prizes are awarded to the scientists who used animals as model. It's quite exciting. And this figure itself says what is the importance of laboratory animal in biomedical research. And currently worldwide, laboratory animal research is $1 million business. A lot of monies, and they are more commercially utilized for the pharmaceutical industry for different purposes. But the authentic laboratory animal house facilities and the certified laboratory animal breeders are countable in our country. I'll show you the figure in the subsequent slides. So as I mentioned already, so I personally feel it is one aspect of the veterinary science that has yet to grab attention. Now, the history of laboratory animal research is not new in our country. So if you go through the history, you see the first institute came, the Institute of Preventive Medicine, Hyderabad, 1870. And they are using animals for plague, smallpox, rabies. Then the Hvac Institute in Mumbai, 1898, plague, cholera, typhoid, rabies, etc. they used. King's Institute of Preventive Medicine, Chennai, the same year, they used animals for TT, cholera, typhoid. Then the Pasteur Institute of India, Kasauli, 1900. Central Research Institute, Kasauli, 1900, and the Pasteur Institute of Southern India, Kurnool, 1907. So it's quite an old history. Now, if you look at the current figure, if you see the CPC ACA registered animal house facilities, there are total 1,687. And you can see the major shares goes to the pharmaceutical colleges, medical colleges, universities, government institute, and research and development institute, and pharma industries, of course. Now look at the figure of the veterinary colleges, it's only 50. And look at the breeder, only 17 breeders, and that makes the figure of total 1,687. Now, there are only 4 major institutions who impart proper training, hands-on as well as theoretical training on the laboratory animals. These are the National Center for Laboratory Animal Science, Hyderabad, National Institute of Animal Welfare, Faridabad, Advanced Center for Treatment Research Education in Cancer, Mumbai, and Indian Institute of Science, Bangalore. So there are not much organizations who actually impart proper training to the professionals or who want to pursue the career or start something in that particular area. Now, if you consider the worldwide total number of animals used, so you can see the contribution of mice and other rodents like rat and hamster they contribute more than 90%. And the other animals are less than 10%. So these are the actual contributor to the laboratory animal research. So I'm extremely happy to note the content of this training, and although it is a 3-day training and it is almost covering all the important area of laboratory animal like usage, handling, genetics and breeding, housing and management, ethics and guidelines, disease models, anesthesia, euthanasia, and preclinical studies. So I'm not going to talk about those things, and in fact, I'm not expert on those things. So my experience with laboratory animals starts when I started my postdoc almost 15 years back, and then I was doing in Virginia Tech, and I had an opportunity to maintain a mouse colony. And then in last 3 years, we are establishing a state-of-the-art modern laboratory animal house facilities in ICR NINP Bangalore, and it cost about 10 crores. So I was involved with the process since from the beginning, right from the planning and procurement, development of the building, and now still it is going on. It's almost finished. Maybe it will be functional within a few months. So I have gathered some experience. If you really want to establish an animal house, so I'll share with you. Now, the biggest question is what kind of laboratory animal facilities I need? Establishing a modern animal facilities is a very, very costly affair. And as applicable for many other things, now maintenance of this, a modern animal house is equally difficult, and it's a very, very cost intensive. Now, when you are doing some research or thinking to maintain some animals, you should be very clear about your requirements. So what is your requirement? Is it about 500 animals per year? If it is so, you really don't need a construct or maintain a sophisticated animal house. It's not required for you. That animals can be maintained at any designated place, maybe at your animal farm or maybe at a part of your laboratory or special room under appropriate conditions and under the standard guidelines. And you can procure the animals from a reliable breeder and source. And you should be very careful while selecting the species or strain for your research. Like your research may need a particular strain. Say if you want to do something on diabetic model, then you should carefully select the proper diabetic model. There are certain strains of mice. It is very difficult to induce diabetes, but there are other mice. It is very easily, very, very easy. Similarly, reproductive model. If you are conducting a research which involves embryo productions or other related reproductive parameters, then you should avoid the strains of mice which have poor reproductive performances. Similarly, immune compromised models. So if your study requires those models, it's fine. But by mistake, if you take a strain which is immune compromised, it can severely affect the outcome of your experiment. So you should be very, very careful. And before starting an experiment, you should go through the literature enough and check which strain or which species is correct for you. Now say your requirement is large and you also have the intention to do some business from the facilities. Say maybe you want to give half of your facilities for the contract research, or maybe you want to breed animals for selling to the other organizations. Then you need to consider constructing a modern animal house. And doing so, these are the few points you need to consider. You absolutely need adequate planning. So you need to be very clear what type of species you are going to use, going to maintain, and what are the number of those animals you are going to maintain there. What are the location of your facilities? Do you have appropriate logistics supports like electricity, water, and others? And what is your plan of the future expansion? Say you have constructed a facilities and after 2 years you realize, oh, I need to maintain another 1,000 animals, and the design of your animal house is so, and that creates a problem. So it should be very, very clear. And most importantly, how much money you can afford. It's very, very crucial. You should decide a facilities that you can really afford. Now, what are the type of facilities? Of course, it depends on the type of experiment you will perform. If it involves just the regular studies, maybe nutritional, immunological, is fine, a single corridor system. But if you start handling with the more and more infectious organism, then you need a double corridor system. So single corridor system is nothing but access to your animal house is through one corridor. So from the same corridor you enter with the clean material, and through the same corridor you come out of the animal room with the dirty materials. Now, double corridor system is you enter to the animal room through one corridor, you take your all clean material and you come out from the other corridor. It's totally different from the first corridor, which is designated as dirty corridor. Come out all your waste material or dead animals or whatever. So this actually helps maintaining a very strict hygienic condition in your facilities, and typically there will be a differential air pressure. So it typically flow from the clean corridor to animal room, then animal room to dirty corridor. And say for our facilities, we keep pressure at 25 Pascal at clean corridor. Then it is the 15 Pascal in the animal room and it is the 5 Pascal. So there is a directional air movement. So there is a very less chance, say from one room to another room, contamination will go, and it helps you maintain the hygienic condition. Then what are the type of experiment you will perform? So there are BSL2, BSL3 and BSL4 facilities, and it depends what kind of facilities you will establish, the type of organisms you are going to use that, and more the BSL level, the more the equipments and facilities you have to add to your existing, your facilities, and that will be, add to the cost. Then you should also plan the adequate storage, like storage for your excess, say cages, say water bottles, say assemblies and other accessories, your feed. And then you should also have adequate service facilities. Adequate service facilities are nothing but like you should have a very good washroom where you will clean the used cages or where you will dispose the used feed materials or the bedding materials, and where you, after cleaning, you'll dry it up before it goes for the autoclave. Then you also should have adequate quarantine facilities, and it also depends how much outside animals, what is the frequency of the outside animals you bring to your facilities. If it is say once or twice in a month, so you really need a small facilities. But if it is once or twice in a week, then you have to think accordingly, because once you put the animals in the quarantine facilities for 1.5 to 2 weeks, it will be there. So you need to plan accordingly. Now, if you are breeding the animals for selling purpose, so it is very important you maintain a very sound breeding record. Otherwise, there might be mix up. You may end up with mixing the strain while breeding, because many of the mice or rat looks alike. And you also have to appropriately train the personals, so who will be maintaining your animals. Now, there are also certain things like equipments. These are nothing but cage assemblies. Then individually ventilated cages. Of course, these are again very costly equipments, and you need to decide whether you want to use those. A autoclave, say a 800 liter autoclaves, whatever we have installed, it costs about 25 lakhs. So you need to decide what will be the size, and typically these are double door autoclave. So it opens one side in the washing room where you dry and wash everything and put in the autoclave, and it opens up in a clean room, which is completely separated from your washing room. And we call it a clean storage, and you take out the material. So you need to decide. You also should have adequate facilities for the deep freezer. So for put your samples or the dead animals, water purifier, you need to have air curtain at the entry or entrance, and of course the air shower if it is a BSL2 and above. So you may also need basic analytical instruments. So kind of a surgical or dissection setup, you may need microscope, centrifuge, blood analyzer, ELISA, and it all depends on your requirement. And typically these are required for the on-site small scale analysis as well as for the regular diagnostics requirement, because if you are maintaining a very big facilities, you have to do the diagnostics at regular interval. I'll tell why. Now, the HVAC system, it is nothing but heating, ventilation, and air conditioning system. So if your animal is very large, it is absolutely mandatory. So it will help to maintain the airflow. It inject the outside fresh air at regular interval, and similarly, it take out the inside stale air through the exhaust, and it helps to maintain the required temperature of 25 degrees centigrade and relative humidity of 55 to 75%. And the noise level of your animal should be less than 60 dB. And now this HVAC system should perform, run for 24 hours. So it based on your electricity supply in your area, you also need to have a power backup. And based on how long your power backup is required, typically genset, you need to decide how much fuel you want to store, how much is the per day consumption. So all this thorough planning is required. Now you also need to establish this very, very standard and strict hygienes and protocol maintaining. So you need to say decide what will be the flow of your material within the say your animal house through the corridors, and you should have a standard operational protocols for all those things, and also the strict standard of animal management, which include feed and water provided, changing bedding materials, disposal of waste. And before that, I mentioned that you need to run a regular diagnosis, because many of the diseases in laboratory animals are actually subclinical and asymptomatic, they don't show. But when you check at regular interval, then only you will come to know that okay, these particular animals is having this infection. And once it is established, it is very difficult to get rid of those. And although you cannot see, but it is definitely going to affect the output of your results. So you should have a very, very clear vision, and it's completely depends on your requirement and affordability. You have to match these two things: requirements, your establishment, your maintenance, and future expansion. You should all have a sound, and you should have clearly defined, and then it should match with the fund available with you, and also the institutional support, how long your institute will support to maintain these facilities. So very fine adjustment is required. Now, I have been given an impression that most of the participants are actually young researchers and students, and there are also persons, young researchers from the corporate and educational institute. So I personally believe the science of laboratory animals and their usage is a huge thing. And of course, 3 days are very, very less, but the course content is so nicely designed, and I'm sure that the participants will get at least the overview and the idea. So what are the things and where they should look into, and if they start with so where they go further, and definitely they will gather much information. And I congratulate all of you for arranging and conducting the training program, and I compliment all the participants for taking part in this. So it is not the end, and it is just the start in the exciting area of laboratory research.
Thank you very much, sir, for a very nice presentation and giving the know-how for establishment of modern animal house, as well as how we can commercialize it and we can use it for different purposes. Then you also in details explained about the various equipments which are required for the diagnostics. So we thank you for your nice presentation, sir. Now this is the time to request today's guest of honor, Dr. Chandrasekhar Raut sir, who also happens to be a proud alumni of this institute, Postgraduate Institute of Veterinary Animal Sciences, Akola. So I request you, sir, to guide the participants on this occasion. Raut sir, please.
Yeah, thank you, Dr. Deshpande, for introducing me. I am really happy to be here, and I would like to congratulate the organizer, the chief patron, Professor Colonel Dr. A.M. Paturkar sir, who is a Vice Chancellor of MAPSU Nagpur, then patrons Dr. Somkuvar, DI and Dean MAPSU Nagpur, Dr. N.V. Kurkure, Director of Research MAPSU Nagpur, Dr. Vety Aher, Director of Extension Education MAPSU Nagpur, and my beloved and contemporary and course director, Dr. Bhikane sir. So I am happy to see you after a long gap. Then Sunil Hazare sir. So thank you very much for coordinating nicely and making this event to start in a good way. And then your knowledge partner Alembic and other course coordinator. I can see here the Dr. Rajesh Limse. So we used to meet regularly for the BOS, and now we are meeting for the another topic, how we can have the proper utilization of laboratory animals. So as Dr. Arindam has explained very nicely in his lecture, how the animal facility would be. So depending on the funds and the utilization of every organization, so one should can have. I was started my career in the same field, laboratory animals, and NIV has given me the very good platform to serve for this field. So I am really thankful to my field, and I am post-graduated from Nagpur Veterinary College in the field Pharmacology and Toxicology. So I could utilize a lot of laboratory animals, and for that purpose, I would like to thank for the NVC also for that matter. And because of that, I was in NIV, one of the national institute, and on that basis, NIV has given me the chance to be a laboratory animal facility in charge. And during that period, I could manage about more than 10,000 animals of the different species: mice, rat, rabbit, guinea pig, monkeys of different breeds. So this vast experience has given me how further we can utilize these animals rationally, not only for the sake of our research, biomedical research, because we should have the utilization properly for the patient's benefit. As I'm in the medical college and to promote the interdisciplinary research. So in the medical college, we are having the different disciplines and focusing on the in the hospital, especially on the patient's wellness. So all this, we have the patient here of different diversity, and once patients are going away or they are discharged from the hospital, so we don't have the material to study in detail. So in that case, laboratory animals are very, very useful. But this sense is very important, how we can have the animal models really are mimicking to the patient's illness. Presently, what we are experiencing since so many years, even in my career also previous days. So if there is an animal model to be developed for the diabetes, but actually diabetes is happening in a different way. It's a metabolic disorder, it's a chronic metabolic disorder. That has a limitations, but in our career or short period, we create the animal model by inducing practices, and we give the streptozotocin or alexon, and within a short period, within 15 days or month, we get that kind of changes, destruction of the beta cells and elevated level of the sugar, and then we define it as a model for the diabetes. So it is not actually mimicking the disease in the patients. So how we can refine it, as we have learned about the 3 R's. So then there are practices or there are concepts. So then we can refine the animal model periodically time to time. Otherwise, we will end up with the false animal model, and we may not give the proper drug development and usefulness of the drug to the patient. So likewise, there are many illnesses or acute diseases, long-term diseases, even the cancers. The cancers, actually we should have the anti-cancer drug and in vitro system and in vivo system together as a triangle. So once we have on the top the patient, and then in vitro system, which will have the link up with the lab animal facility. Everybody cannot do everything. As Dr. Dhani has suggested or described the laboratory animal facility, how one can have. But in my opinion or my views are there, everybody can have the very good lab animal facility with their defined system, whatever they have. Only thing, they have to record each and everything very nicely proper. For example, any disease in the animals who are handling any kind of species, maybe small animals or large animals. So generally in the MOPS, we handle the many cases in the hospital, the farm animal or domestic animal. So if these animals are similar to the or close to the human diseases, so that can be defined as a natural animal model towards the human disease. So how we can come to that closeliness? So one should have the all parameters to be documented properly. Then guidelines are there. Guidelines are just guidelines. There is a lot of scope to do whatever we want. MOPSU would like to do anything, so they can do independently. They can develop their own institutional guidelines under the MOPSU, and it can be then communicated to the CPCCA because practical problems are different. Theoretical problems or theory is different. So likewise, everybody can contribute throughout the nation. So with this small small talk or small words, I would like to end this talk and I would like to thank the coordinators and the patrons and the MAPSU authorities. So thank you very much for inviting me for the guest of honor. Thank you very much.
Thank you.
I'll be there to participate with you and any queries. I am available always to anybody.
Thank you, sir, for your kind words, and whatever indications you have given, we'll take a note of it. We'll try to start it at MAPSU. And already Dr. Hazare is doing a lot in the same field. Dr. Hazare is also arranging training for this Ayurveda college students, and other things are also going on. So thank you, sir, for your nice words.
Thank you.
So now I would request the chairman of today's function and our beloved associate dean, Professor Dr. Anil Bhikane, sir, to address on this occasion. Bhikane, sir, please.
Thank you, Kuldeep.
Thank you, sir.
Chief guest of inaugural function of National Online Training Program on Laboratory Animals in Biomedical Research, organized by Postgraduate Institute of Veterinary and Animal Sciences, Akola, Dr. Dhali, who is presently working as the principal scientist, National Institute of Animal Nutrition at Bangalore. The guest of honor and our old friend, Dr. C.G. Raut, who is working as the director, Interdisciplinary Approach, D.Y. Patil Medical College, Pune. Course coordinator of this program, Dr. Sunil Hazare. Course coordinator, co-coordinators, Dr. Kuldeep Deshpande, Dr. Limse, Dr. M.K. Patil, Dr. Karande, Dr. Uma, Dr. Burekar, and participants. At the outset, I welcome and thank the chief guest of today's program, Dr. Dhali, who spared his time for this inaugural function. I'm really thankful to Dr. Dhali, for his elaborate presentation on how to establish the modern, you can see the Laboratory Animal Research Center or animal facility as such. He has shared his own experience with the participants. I'm also thankful to Dr. Dhali for sharing a very good information in his inaugural speech. Really, we came to know that 73 Nobel laureates, they have worked on the laboratory animal models. That's interesting information he has shared. Thank you, sir. I was also not aware that in India, this laboratory animal facility was present and it was being used since 1870. Another interesting information he has shared that there are 1,687 institutes that have registered with the CPCA, but there are only 50 institutes, veterinary institutes, which have registered with the CPCA. And ours is the one of the institute which is organizing this program. Dr. Dhali has shared his experience as regards the establishment of a wonderful, you can see the modern type of a laboratory animal facility. According to him, this is no doubt a very costly affair. But Dr. C.G. Raut in his speech told that even a small, you see the laboratory animal facilities can be established at many places with the available resources, on that too on a smaller scale. So many of the institutes, they are having the laboratory animal facility, maybe on a smaller scale, but definitely they are using these facilities for the research purpose. I'm thankful to Dr. Raut sir for his gracious presence on this particular occasion. Actually, I could see him after a gap of 10 years. I still recollect that memory. He had been at with Geer for delivering a lecture in a physical program. Today, he is available here in an online type of a program. And we know that Dr. Raut sir is having a vast experience in the field of laboratory animal research as such, laboratory animal care and management. And he's one of the guest faculty and a speaker in this program. And I'm sure that his lecture will be wonderful. It will be very interesting for the participants. Dr. Wangikar is also participating in this training program, and he's also having experience, and he has developed his own, you see, the facility and a contractual type of a research facility. So I'm sure that he will also deliver good, and that will be very important and interesting for the participants. Even the other speakers, they will also share their knowledge and experience with the participants. Dr. Dhali has rightly pointed out, this is you see, a very short type of a training program, maybe of a 3 days duration, but really important in this current scenario, that the laboratory animals, Dr. Hazare rightly told that this pharmacy is developing very rapidly in the India as such, and it is a very good hub for a pharmacy. And we all know that the drugs, they are being developed, drug development in the area of drug development, the drug trials, and as such, studying the drug toxicity and all these things, the laboratory animals, and they are very important. And there is a lot of scope for the budding vets, as well as the graduates and postgraduates from the life science students in this particular area. And this particular Dr. Arindan Dhali has also rightly pointed out, when we were a student, the actually laboratory animals, the different aspects of the laboratory animals, they were not included in the syllabus. And but in the last 10-15 years, definitely the BCI syllabus includes some topics on the laboratory animals, and our students are getting some exposure. But the basic aim of this training program, designed by my colleague Dr. Hazare, is that the students, particularly the student fraternity from the veterinary colleges, as well as from the life science branches and the institutes, they should get at least a preliminary knowledge about the laboratory animals, how the laboratory animals, they are being bred, how they are to be fed, how they are to be managed, their health care, and the rules and regulations as far as this, you see, the formulated by the CPCA. And I hope the speakers will definitely cover all these aspects, and the deliberations in this particular training program will prove useful to the participants. I, on the behalf of PGIVAS, I see the welcome the participants from the nook and corner of India. 190 participants, they are participating in this program. So that's a very good thing. And I congratulate Dr. Hazare and team for making efforts in organizing this program. I know my colleague Dr. Hazare is taking pains for the last 1.5 months for organizing this program, and I hope that his efforts will prove fruitful by disseminating the at least basic information about the laboratory animals to the participants. Once again, I thank the university administration for granting permission, and our chief guest, Dr. Dhali, and the guest of honor, Dr. Raut, for participating as a chief guest in the inaugural function of this training program. I congratulate the organizers, particularly the coordinators and the co-coordinators. Thank you. Thank you.
Thank you very much, sir. Thank you for your kind words and sharing how the genesis of this training program was done. And now this is a time for a formal vote of thanks. And I request the co-coordinator of this training, Dr. Rajesh Limse, sir, for offering a formal vote of thanks. Limse, sir, please.
Thank you, Pandey. Dr. Hazare, sir, thank you very much for giving me this opportunity to express a sense of gratitude towards everyone, each and everyone who is associated with this wonderful training program on laboratory animals in biomedical research. At the outset, I would like to thanks our beloved Associate Dean of Chairman and Associate Dean of PGIVAS Akola, Dr. A.U. Bhikane, sir, who is a dynamic leadership and always a source of motivation to the staff for arranging such type of wonderful programs. Then I'd like to thanks Dr. Arindam Dhali, who is chief guest and principal scientist at NINP Bangalore, to whom just now we have listened, and hope many interesting things come out of his wonderful speech, short speech. Then I'm very much thankful to Dr. Chandrasekhar Raut, sir, who is Director of Interdisciplinary Research at Dr. D.Y. Patil Medical College and Research Center at Pune. And I'm very much fortunate to have opportunity in the past to work with him during as a member of our Board of Studies. And really, he is a very enthusiastic and person, and every time on the mission, and many things are there to learn from him. I'm very happy that he is associated with this training program. Then it's time to say thanks to Dr. S.W. Hazare, sir, who is a course coordinator and taking a lot of efforts along with the course coordinators, including myself. And I'm really thankful to him for being instrumental in arranging such wonderful training program. Then we are having a list of some guest speakers who are expert in their field, like Professor Dr. Pritam Mohan, Dr. Vikas Dikhe, C.G. Raut sir is there, Dr. Bangikar is there, Dr. Arvind Ingre sir is there, Dr. Panjali Pure, then Dr. Girish Panchabhai, Dr. S. Sole, and Dr. Bankar. I am thankful to you all for giving your valuable time while addressing audience. And I'm highly thankful to the knowledge partner Alembic Pharma team, Dr. Santosh Shinde and Mr. Karuna Nidhi for this wonderful training program. And at the last but not least, I would like to put my sincere thanks to the all participants who are here as an important gathering of this training program and instrumental in making it a huge success with their keen interest. Thank you very much.
Thank you, sir. Thank you for the formal vote of thanks. And now this is the time to start the lecture first. So with the permission of Chairman, I would request the first speaker for this training program, Dr. Sushant Sole, Assistant Professor, Department of Pharmacology and Toxicology, Mumbai Veterinary College, Mumbai. I would request Dr. Hazare sir to give introduction of today's first speaker. Hazare sir, please.
Dr. Sushant Sole is presently working as an assistant professor at Mumbai Veterinary College, Mumbai. He did his graduation in the year 2006 from Bombay Veterinary College, Mumbai. He did his MSc from Chaudhary Charan Singh University, Haryana Agriculture University, Hisar. He did his PhD in year 2012 from University of Delhi. He has got more than 8 years of experience in the field of drug discovery and in the academics. He was scientist at Sriram Institute for Industrial Research and New Delhi. He worked at Sun Pharmaceuticals, India. He also worked at Denmark Pharmaceuticals, Mumbai, as a project manager, drug discovery, research and development. He has handled many projects and he has guided around 8 PG students, and there are 12 international and national publications to his credit in high impact factor journals. So this is all about Dr. Sushant Sole, and over to Dr. Deshpande.
Now I request today's speaker, Dr. Sushant Sole, to share his screen and please start his lecture.
Am I audible, sir?
Yes, you are audible.
Just a minute, I'm sharing the screen.
Okay.
Honorable Director and Associate Dean, Professor Bhikane, sir, course organizers, Dr. Hazare, sir, Deshpande, sir, Limse, sir, and all respected team members of the committee. First of all, I'm very thankful to the organizers who give me an opportunity to address such a big forum, and really indebted for it. I will be starting my topic today. It's introduction to laboratory animals and handling of animals. The most of the slides are prepared in the pictorial forms so that the students will get an easy access to how exactly the work is going to happen. See, all of us are quite aware about the nature of the human being, that we are continuously being encountered with different disease elements. And in order to understand that disease element, we must understand its basic process, what is happening at cellular level. So there is an utmost need to understand this basic need to conduct the experiment on animals. And there's a need of animals comes into the place. So if we consider the use of animal in research and education, it has the use of animal into the actually research and education has started long back. Maybe from the time of you can say the humanity has started onwards. Because in my next slide, I will show you the some pictures where the use of these animals are extremely important while developing the different therapeutics. So there was one you can say in 2007, a Russian actually space scientist has used this Jabrin as an experimental animal into the spacecraft. They have studied the different behavior of these animals before and after into them. So just to make you aware how the vast use of these animals can be there into the different fields other than the pharmaceutical research and all this. So as far as the statistical data is concerned, how much animals are being used actually into the research, almost the extensive use of the animals is being there into the toxicity studies, especially most of the new compounds or new molecules which are there into the industries are being tested on these laboratory animals. And the range of the animals is not only limited to rodents, it can be from your Jabra fish to the higher species of animals, right? So I hope you understand the animal experimentation. What is the animal experimentation? It is nothing but to mimic the condition of your end users or end disease process we induce the particular disease elements into that animals and conduct the different procedures, analysis on these animals, and to understand how the disease process is being done. And why there is a need to understand this disease process? Because we need to target that particular disease, particular therapeutic molecule. See, the term animal testing, animal experimentation, animal research, in vivo testings, so all these are interchangeably used, and especially the VV section. This term is basically used by the people who oppose the use of animal into the research, which involves the cutting of the dissecting of the living animals. So as far as data is concerned, I was talking about this, almost 50 to 100 million animals ranging from Jabra Fish to non-human primates are being used every year. So this much is the huge potential of this field. So few questions I have pointed out. Why do we need the animals into the research and education? The answers before my this talk, I think most of the speaker who has talked about the importance of the animals into the research has given the clue about this question. So we need to understand the disease process, and in order to understand disease process, we mimic the condition into the animals, and therefore, and as far as to safeguard our human population, we conduct the experiment into the small animals. So can't be the computer models and cell cultures replace the animals into the research? So this is another question. So why not we completely rely on these computer models and cell cultures? Because nowadays this concept is coming into the place that even in most of the industries are also being using these different computational models to minimize the use of animals into the research. So here what they do, they based on the available literature or the based on the data available into the public domain, the molecule which is being new to the pharmaceutics. So in that area, they try to evaluate the effects of that compound based on the structures and give the prediction about that molecule. How much will be the toxicity and how much will be the chances of the death, or how much will be the efficacy. So again, it has a limitations because it cannot exactly give you the disease condition process, how it is happening at cellular level. Right? So can results from animal studies really be applied to humans? So this is a very interesting question, because why we cannot completely apply all the animal data to the humans, because it depends on the translational value of that particular research. For example, we are always talking about this diabetes. So even though we can produce the type 2 diabetes or type 1 diabetes into these animals, it is not exactly how the human diabetes mimic that process, right? Because the process of regeneration of the streptozotocin induced pancreas or by different drugs, whatever we are inducing the animals, it start regenerating themselves. Even these species are quite sturdy. They again try to regain this. So the best, the best you can say the example for this is when you induce the diabetes into the animals, not 100% of animals will show the high glucose rate. We have to screen it because most of the animals will oppose that particular disease process. So these are the few discoveries which has happened in last couple of years. In 1901, so long back, the diphtheria toxin, it was introduced into the with the help of horses. Actually, and the scientist's name was Emil Edorf. In 1922, insulin. So insulin was by scientists Banting actually, Banting and that. So he produced the insulin with the help of dogs as a model. Then prontosil, we can call it as a sulfonamide prodrug, and it was the first drug of choice, you can say, for different bacterial infections. And the major discovery was been done by this dogma scientist, and he got a Nobel Prize for it also. Then penicillin, all of us are quite aware about the Alexander Fleming discovery. It has proven his records into the rat species of disease. That disease process that he has mimicked into the rat. Then Waxman, he introduced the streptomycin efficacy with the help of chicken. Again, the polio vaccine. So Landsteiner and Popper has proved the infectious nature of this disease with the help of these animals. And later on, the vaccine was developed by the scientist Sark and his co-worker. So by considering the different huge area where these animals can be used into the research, I have prepared the slides just to make you how the animals are being handled, how the blood collections are being done, what are the different strains of these animals and all. So let's first start with the mouse. The kingdom is Animalia, and phylum is Chordata, class is Mammalia, and order is Rodentia. So mouse species what we call it as mus musculus. So there are different types of this mouse strains. I have pointed out very few mouse strains. One is bulb C mouse, then SCID mouse, then albino is the most commonly used strain actually, and CD1 mouse, then CL B6 mouse. Then there are nude mouse where the thymus is absent in the animals by using the different drug-induced method of destruction or by using gene manipulation. So these are the different strains where the animals can be used for producing particular disease. Knockout mice. See, this is the clear difference. You can find it out how the obese OB genes by altering the OB genes, these animals are being produced. So obese mice is there. Then transgenic mice. Here the bioluminescence can be seen into these animals because by labeling the particular luminescent into that gene, we can look at the behavior of the animal by this fluorescence. So as far as biological data is concerned, these animals range between 15 to 40 actually. So biological gestation period is 19 to 21 days, and average lifespan is almost 1.5 to 2.5 years. Litter size is 5 to 7, and it may be a more based on the strain and the time of parturition and all. Estrus cycle, it's a 4 to 5 days. So blood collection method here, the what are the requirements for the blood collection? So that 75% alcohol will be common for most of the cases, and small plastic bottles for half centimeter diameter bones, then scissors, pipette, and tips and vial for the collection. See how the restraining of the animal is being done in case of mice. So there are a number of ways you can restrain by using the restrainer. Also, we can restrain the animals, but the best way to handle the animals is put your hands on the neck of the animals by holding the tail of the animals. Basically, this process has to be done on the cage grills because it's easy to take the grip of the animal on this surface. So this is a restrainer where the animals can be restrained by using proper restrainer. Here it's a transparent restrainer where there is a small hole on the tip of that one point where the animal can breathe easily, and the tail portion will be out. You can perform the experiments on the tail by using this restrainer. So this is another leave the tail of the mouse outside and cover the restrainer. So either if you want to do the experiment on tail, then this is the best restrainer we can say. Another approach is you can put the animal inside the cage and take out the tail of the animal from that cage top actually, and you can perform the experiment on the tail directly. So there is no need to use the restrainer in that case. This is the tip, how the tip of the tail can be amputated here, because such kind of requirement is there during your experimentation blood collection method. When the blood collection through IV route, through intravenous route and through your retroorbital is quite difficult, and you need very small quantity of blood. In that case, you can amputate the tail and collect the blood. So by using the micro pipette or pipette, you can collect the blood into it. So orbital sinus. The next approach is the collection of blood from orbital sinus. So here, first of all, this is very painful procedure, and we need to apply the anesthetic before blood withdrawal. A convenience and easy apply method for blood collection in mouse because we collect the maximum amount of blood by using the orbital sinus. Almost 0.5 ml we can collect by. So what are the requirement as always? 75% alcohol and 27 gauge needle and 1 ml syringe. It can be used for collection of this, because syringe, why? Because to anesthetize the animal, and glass capillary tube. So tubes, capillary tubes can be of variable sizes also, and just to keep it handy, make it half portion, whatever the market capillaries are available here, you can make it to half, so that it's easy to drop the blood into your pipette. It can be coated with the EDTA also. By taking the solution of EDTA, you can put the capillaries inside and take the EDTA blood if you want to work on the plasma. And if you don't want to work on plasma, you can directly collect and blood and work on the serum. So this is the way how you anesthetize the animal, or how the intraperitoneal injection can be given. So basically, you have to hold the animals in such a position so that the internal organ should be directed downward. Because if you are putting the animals in this fashion, the organs may comes into direction of your needle and may rupture the organ. So you have to pull the animals in such a way that the organs will be directed downwards, and there will be a complete vacuum you can feel after injection of the needle, right? And just to ensure that it is not into any organ, you can slightly take it out. You can slightly withdraw the small amount of portion from peritoneum, and you can see the normal bubbles will be there. And if it is if the blood is there, you can make sure that this needle is being inserted into the organ. So this is the way how the retroorbital is being done. See how by using the sharp end of the glass capillaries. Okay, so you have to insert at the inner canthus of the orbital and penetrate it in such a way that there is rupture of the capillaries inside that ocular, and the blood will be started oozing out very fast. Here, what we have to take care of it, you should not damage the eye of the animal, because while inserting the capillaries, sometimes the internal canthus or the capillary glass may get stayed there during the rupture. So make sure that any glass material should not be there into the inner canthus, because the animal may try to struggle it if it is not in anesthetized state. And or if it is recovering from that anesthesia, you will try to take it out all the capillaries and may damage the eyes. So this is way how to collect the blood. After putting by capillary action, the blood will start coming into the capillaries, and you just have to put it into your peritoneus. So blood collection from cardiac puncture. So this is also one of the methods we used to do into the industry. So, but it needs a lot of expertise by doing the blood collection by using cardiac puncture. We have to literally palpitate the heart. So maximum 1 ml of blood can be collected by the sweat. Usually such kind of collection is being done at the end of sacrifice. So when the animal is being sacrificed, at that stage you can collect the blood from this cardiac puncture. What are the requirements here? It's a 27 gauge needle, 1 ml syringe for infusion, and 24 gauge needle is almost 3 ml syringe for blood withdrawal. So first of all, you have to anesthetize the animal by using the injection. So we can use the different anesthesia. I think there is another lecture on the anesthetics used in animals. So basically, we can use xylazine ketamine combination. We can use the thiopentone. So or if you are using it for small time, you can use the ether and diethyl ether or the cyclopropane. So dissect the thorax. With this, you have to clean the area by using the 75% alcohol by using the cotton ball. And you can give the artificial ventilation here by using the different aerators. First of all, palpitate the heart and see how will be the direction of your needle. It has to be perpendicular to the surface, because if you are making it into a 45 direction and all, it may pierce the diaphragm and most of the other organs also. It has to be perpendicular, and look at the palpitations of the heart first of all, and then only you should insert the needle. The depth of the needle can be seen based on the actual experience only. So first few couple of times you may insert it till the vertebra directly. So that's how it has to be very peculiar. Hardly a 0.2 or 0.3 mm first you have to insert, then slightly try to collect the blood from that area. And if it is coming very easily, first hold on for a few seconds. Again, during the contraction and relaxation, the next blood will come into the syringe. So this is the withdrawal process, how the process is being done. See, the ventilation is being given by artificial route. Blood collection from saphenous vein in mouse. So this is the method used of multiple samples. When you have to take multiple samples, then you can try this method. And basically IV studies and all can be done. So rats, hamsters, jabrils, and guinea pigs, in all the animals, this method can be applied. So what are the tools for it? Again, 50 ml syringe tube with small holes at the end of the retainer, then scalpel and shaver just to remove the small hairs on the skin, then needle for release of blood, then tips and pipettes for blood collection. So again, you have to restrain the animals. How the restraining is being done, I think in first couple of slides it is more clear. You have to hold the tail first by using your left hand, and then hold on. By using your right hand, and by using your left hand, you can hold this neck of the snake. So if you are using the restrainer, you can put it here. Shaving can be done into the respective area by using the ready shavers, or you can use the normal shaver also. So it has to be cleaned very properly, because this hairs will not it will make you difficult to see the saphenous vein into the same. So it's very clear here. Saphenous vein is seen on the surface of the thigh. Thigh surface will be very clearly. Apply Vaseline after disinfecting the surface area to reduce the clotting and coagulation during the blood collection. So we have to make sure sometimes that vessel is not clear. So just to raise it, we apply alcohol. Sometimes xylene is also applied. But these are very crude methods. Xylene will cause severe irritation to the skin. So try to avoid it. A 24-gauge needle is used to puncture the vein and release the blood from saphenous vein. So you have to insert very slowly, so that because if you are not able to insert properly, it may cause the collapsing of the blood vessel, and next time you won't find the blood at that time. So this is how the collection of the blood can be done by this saphenous vein. Approx 100 microliters can be collected of the site. So flex the foot of the mouse to reduce the blood flow back into the puncture site. So just hold it into a downward direction, right? And cotton ball can be applied to reach the next oozing out of the blood. So intraperitoneal injection, I already shown you this approach. Subcutaneous injection is also easy. You have to hold on the skin fold of the neck region and insert the needle into the. A lump of substance can be seen through the skin if you are administering as an intradermal site, or even subcutaneously also. You can see the complete lump onto the surface area. If it is shaved area, you can easily find it out. Now, this is very important aspect: oral feeding of the mouse, so or rats, mouse, whatever it is. So, in general, this approach is being followed into the industry. Most of the compounds are being tested by this oral route, and we must ensure that the administration is very proper, because while administering the compound by oral route, the chances of death are quite high, because you may administer the compound into a lungs directly. So, it has to be very meticulously done. So how much maximum amount can be done? This is so it's a very common question the student may ask. See, if it is rat max, 2 ml of blood, 2 ml of volume of liquid can be administered. So again, it depends on the body's weight and the size of the animals. Usually 1% of the body weight is the feeding volume. The needle size which is used here is 18 gauge stainless steel needle, and it has very typical shape, slightly curved at the top of the needle, and ball is being there at the one end of this needle. Okay, so you have to use the gloves to restrain the animals properly. See how the administration grasp the loose skin on the back of the mouse. Okay, the fold of the skin has to be in such a way that it should not be too tight and it should not be too loose. See, if it is too tight, then it may strangulate the animals and animals may die because of the choking. So it has to be very, very gentle also. And again, if it is too loose, the animal will struggle a lot and will not be able to administer the drug. And even if you try to struggle it with the animals, sometimes the animal may die immediately because the blood because the liquid or the oral material what we are administering may enter into the lungs territory. Okay, so usually on the left side of the your animal is being given because you can easily experience that smoothness of administration. If it is into the lungs, you will encounter the lot of struggle, and the animal will try to gasp if it is going into the lungs. Right? So this is the identification test if it is going into the right direction or not. Sometimes there is a need of the experiment that it should directly go into the stomach, or sometimes it should go into the pharynx, or sometimes it should go into the esophagus. So based on that requirement, you should decide the depth of your insertion into the mouth cavity. So this is most of the methods what I have. Sorry, am I audible? Hello, am I audible?
Yes, you are audible, sir.
Oh, okay, sorry, just there was a call in between. So see, most of the method which we have seen in mouse can easily be applied in case of rats. So I'm not putting the separate pictures for the rats here. So there are two main strains which are commonly used into our research and education purposes: the Wistar and Sprague-Dawley rats. So Wistars are quite resistant to infections. They're quite sturdy animals, and so but tumor study and all, it's usually not preferred because they have very low incidence of tumors. It has a wide head and long ears, so tail length is usually less than your body. In case of Sprague-Dawley, rapidly growing strain is there, so longer and narrower head, pointed with the head, and longer tail is there, and decreased respiratory infections. So the respiratory infections catching capacity of these strain is very high. It is very low. So most of the respiratory studies are fully avoided in this places, in this strain. Now coming to the handling of rabbit. So we are going to study these points here: how the intramuscular, subcutaneous injections, blood collection, and sexing is being done in animals. Right. So this is the exact way how the animal has to be handled properly, because you should not feel stressed when you come across his habitat. See, normally when we used to work into the industry, especially when I was in the Daiichi Sankyo, it's a Japanese firm, which was being known as Ranbaxy. So Ranbaxy was taken over by Daiichi Sankyo. That some Japanese scientist has given very nice actually acclimatization technique. Actually, they have taught us the technique. So whenever we used to start any study, first 15 days we used to acclimatize the animal. It's a normal process of acclimatization. But they used to ask us whosoever the scientists are there will be working into that particular model. So these scientists has been asked to be with the animals for a couple of time, or they used to handle the animal with very gentle approach, so that the animal will feel the very friendly nature with the scientist who will be working with it. Because the stress which will be there by putting the animals into your hand, that can be minimized by using that acclimatization technique. So in that technique, we used to hold on the animal, and they they used to the animal used to make it comfortable actually. So this was the technique during the I was in the industry. They used to taught us how the acclimatization is being done to the animals. So this is the insertion, how the insertion of the needle should be there into the skin. And in case of rabbit, the hairs are quite high, so you have to make sure it should enter it at the dermal region properly. Subcutaneous injection is the most common method for immunology study. So we prefer the animals, rabbit, for the immunology testing. The 21 gauge needle and 1 ml syringe is preferred for this kind of study. So initially, what we have to do is to disinfect the injection site by using the 75% alcohol and pinch the skin. So here, first you have to anesthetize the animal also. Keep the needle deep in the skin for a while after injection to prevent the leakage substance from the needle, which due to imbalance of pressure emulsion inside the syringe. So what does it mean? Sometimes because of that pressure, when you administer the needle into that region, it may causes the oozing of that fluid outside that inserting point. So just hold on with that point for some seconds, and then you have to administer the liquids. So collection of blood from ear vein. So usually very small amount of blood can be collected here, and we have to use sometimes tranquilizers because the rabbit may struggle a lot. Or there are peculiar restrainers available, rabbit restrainers are available, where we can use the animals without using any tranquilizers. But ethically, we should not cause any pain to the animals, and we should use the tranquilizers or anesthetics for collecting such a blood collection, or maybe any surgical approach if we are doing. So ear vein is preferred in the rabbits for collection or administration of the drug. So see, this is the clear picture how it looks like. So you have to make clean this area first by using any liquid or by using the foam. You can clean that particular vein, and this vein is very clearly visible. Usually marginal vein is preferred because it's quite easily detected. It can be easily administered also. The flow or the resistance is quite low in that case. So fix the position of the needle by your left thumb and withdraw the blood from here. Okay, and immediately after removing the needle from the insertion site, you have to make sure that cotton plug should be applied immediately at the injection site. Okay, apply the clip. Ideally you should use the foot of clamp. Okay, you can hold on with the help of the cotton plug. So whole blood collection from carotid artery. So this kind of experiment, when I was in industry, we used to do it. Usually blood pressure and all experiments which are related to your heart rate and all. So these experiments we used to do by using the carotid artery cannulation. So for this experiment, you should make sure the animal is in general anesthetic, and make there are chances that the animal may revive. So make sure the animal is in continuous stage of general anesthesia. The chances of mortality is very high in this case, but the recovery rate will also depend on the depth of the anesthesia. Right. So apply some procedures for collecting blood and all. So either you can give the intravenously anticoagulant. Suppose you are doing an intravenous study and all. So what are the tools? So electric clipper to remove the hairs, then cleaning, scalpel, tissue forceps, all the surgical equipments what we need here for carotid artery cannulation. Okay, the surface area where the cannulation is being done. Okay, where the animal is being put into that surgical table. So ideally, that ideal surgical table is having the lamp, and which make it hot. Actually, that surface usually being made hot, so that the animal should not feel shivering during your anesthetic recovery and all. So you have to shave the your neck region. See how the shaving is being done by using the electrical shaver. You can do it by using your normal blade also. Usually neck region below your pharynx, so almost 5 centimeter opening on the middle line of the neck has to be done. Okay, See, this is a clear picture where you have to remove the fascia and you have to find out the carotid. It's a whitish color structure, can easily be noted. Usually it is accompanied with your jugular vein also. So you have to separate the fascia and find it out the carotid. Once you find it out the carotid, you have to hold it by using the forceps upstairs, and then take a small nip onto the carotid and then inoculate with your cannula. Right? So this is how the insertion and the ligation is being done by using the carotid artery. So this is the model usually being done to study the blood pressure and all, where the manometers, one end is being placed here using the carotid artery measure. Okay, sexing. So this is the most common method. So you have to slightly apply the pressure on the external genitalia to expose the vulva of the female body. So you can easily find it out. And slight pressure is applied to the external genitalia to expose the penis of the male. Okay, so in case of penis, you can find out the clear difference here how the male and female can differ. Sometimes you can easily note down the testicle also if you are pressing it here. Okay, the next species is guinea pig, Cavia porcellus. So these are very you can say docile animal, and most of the TB or anaphylactic work or any sensitivity testing, skin sensitivity testing, so all kind of that experiment is being done preferably on guinea pigs. So highly sensitive to histamine and penicillin, and require the exogenous vitamin C diet. So you may ask the question, why vitamin C for this species? The reason is they cannot synthesize the vitamin C. They lack one enzyme for their synthesis. So biological data, typically adult weight range in between 600 to 800 grams, and average lifespan is almost 3 to 5 years. Gestation period is 68 days, and average litter size is 3 to 4. So where we can use these animals for using different respiratory, as I said, for the respiratory study, we prefer these animals for bronchodilator activity, then anaphylactic activity, anaphylactic shock and all. If you want to induce the animals, these are the most preferred species because the manifold higher response can be seen into these animals. Okay. So Bioassay of digitalis, because the cardiac size is quite high, and cardiac size is quite big, and we can easily evaluate the different parameters by using the guinea pigs. So what are the ways to collect the blood? By using the marginal ear vein and cardiac function. See, this is how the there are certain anatomical differences I have pointed out. So usually they have 4 digits on the forelimbs and 3 digits on their hind limbs. And the ears of the guinea pigs are small and accessible to vein access is very difficult in this species. And guinea pigs have very large tympanic body, and the internal structure of the ear are easily reached features. So that makes us to work on the auditory research on these animals. If you want to work on certain temporal ones or auditory reflexes and all, so these guinea pigs are preferred here. So both male and female have a tits. So female has a single pair of inguinal mammary glands. So the thymus of the guinea pigs surround the trachea. This is in contrast to your rat, mice, and other muride rodents, in which the thymus is deep within the thoracic cavity, so overlying the part. So guinea pigs are monogastric animals, and they have lower intestinal tract, typical of herbivorous, and the large intestine occupies most of the abdominal cavity. Female guinea pigs can be distinguished from the male by a shallow or U-shaped break in the ridge. So here you can easily find out the difference here into the external genitalia. So this break is covered with a vaginal closure membrane, which is only open during the distress or parturition. Everted penis. So here the sexing is very easily done by applying slight pressure. You can evert the penis of the boar and find out the male by using the technique. So subcutaneous injection, neck region, as in case of rat, mice. Here also we can give it into the neck region. Okay. The intravenous injection, marginal ear vein is possible but difficult due to small size more difficult to classify. And other sites like saphenous vein and lingual veins are preferred. See, there's one more approach, but it's extremely painful approach and usually not recommended. But sometimes it's very difficult to find out the veins in these animals. So penile region is being preferred. So dorsal penis vein is quite visible in this animal. You have to press the penis out, and you can give this injection into the veins. But as I said, it's a very painful. We have to take the pre-medication. So anesthesia is very must for this administration. Okay, coming to the next slide. So I'm a little bit fast because I need to sum up all the slides. So Jabra fish is another strain nowadays. It's very popular into the research. So these are the pictures from our laboratory. One of my students has recently finished this work. So Danio Rio is the species. We have used Jabra fish for the endocrine disruption potential. So embryo, larva, and adult, these are the pictorial representation, how they look like. So usually for high throughput screening, these animals are being preferred. What do you mean by high throughput screening? When the large number of molecules or very huge number of molecules are being generated in your chemical lab, then if you want to test it for at the same time to save the time and all, we usually use the low cost animal where the and short lifespan animals and preferably Jabra fish. See, these are the pictures from my students' thesis. Normal embryo, how it look like. So usually larvae at the 48 hours of your post-spawning, and how they look like. See, very clear structure it can be seen in these animals, and therefore these are the most preferred species nowadays for most of your nervous studies. So Jabra fish is an ideal model for organism research nowadays. Was utilized as a biological model by George Steiner. He was one of the scientists in University of Oregon in 1970s. So it is highly validated as powerful vertebrate because it has a maximum translational value. As I said, 70% at least it has a maximum translation value, which is in contrast to your rodents. Okay, because of the genetic makeup of these animals. They possess external fertilization, and their development pattern facilitates the observation and experimental manipulation of the embryo. So you can, because the transparent membrane is there, you can easily look into the animals, into the embryos, and see how the development is being there. So moreover, they are large cluster size. Size is almost 200 to 300 per fish. So the data generated by your testing is very huge, and therefore precise decision can be taken on the basis of your data. So one of the most unique advantage of Jabra fish is unrivaled optical clarity. So as I said, iska jo membrane hai, it's very clearly visible and allowing the visualization of individual genes. So fluorescently label kar sakte hain genes ko, and or dye label bhi kar sakte hain. You can use that dyeing techniques to see how the embryo looks like. So due to the small size of larvae, high throughput screening and neuroactive compounds can be easily found. Okay, the embryo and chorion protective membrane surrounding of the embryos are transparent, and during the first 5 days, the major organ system develop. Okay, so or even start functioning also. So all the new degenerative changes, if it has caused by your drug, can be easily seen by using the Jabra fish. So it has proposed a better model system than the rodents, as they allow in vivo analysis without disturbing the physiological milieu of the disease. So there is no need to alter any other physiological process. So we are continuously using this model for different endocrine disruptions since last couple of years, when almost 6 to 8 years back, I think during our ex-professor's time, it has started this model. This is in short about the presentation. Your questions are welcome. Thank you.
Thank you, Dr. Sole, for extensive presentation for your topic. You have nicely presented your topic, that is introduction and handling of lab animals, especially various sites you have shown. So though this is not theory as well as practical type of training, but by seeing the pictures and exact position of things, so it was nicely explained by you. that how these things can be taken care of. And we thank you for your nice lecture. So the questions, if ever they are there, so I request the participants to put their questions in the WhatsApp group because we are lagging behind the time. It's almost 4:44 now, and we are waiting for the next lecture. Uh, Dr. Pritam Mohan is there from Khanapara, Guwahati, Assam. Dr. Mohan, sir. So I welcome Dr. Mohan, sir, who is a professor, Department of Pharmacology and Toxicology, College of Veterinary and Animal Sciences, Khanapara, Guwahati, Assam. And I request the coordinator, Dr. Sunil Hazare, sir, to give brief introduction of today's speaker.
Professor Dr. Pritam Mohan is presently working as faculty of Veterinary Sciences, Assam Agricultural University, Guwahati, Assam. He did his master's in the discipline of veterinary pharmacology and toxicology from Assam Agricultural University and PhD from Indian Veterinary Research Institute, Ajit Nagar, Bareilly in year 2000. He is my PhD classmate. He did PhD. He has teaching experience of 29 years and guided 12 MBSc students, 2 M.Pharm students, and 2 MSc students, and 5 PhD students. He has published 25 research articles in national and international journal of repute. He worked on 3 extramural research projects. He is a guest faculty at National Institute of Pharmaceutical Education and Research, Guwahati, and visiting fellow at Dibrugarh University. He is academic counselor at Indira Gandhi National Open University. He is consultant at Laboratory Animal House, Institute of Advanced Studies in Science and Technology, and he is CPCSA nominee at Animal Welfare Board of India. He is on the board of management of Assam Agricultural University. He is a member of governing body Girjananda Institute of Pharmaceutical Sciences.
So we welcome such a learned speaker with a vast experience in this field. So I request you, sir, to start your presentation. Please share.
Yeah, sure. Thank you so much. It's nice to be with you people all the way from Guwahati and have a audience throughout India. It's really good. So I'll be starting my talk very soon. Let me share my presentation. Okay. Is it visible?
Yes, sir. Yes, sir. It is visible.
Okay. Already the previous speaker has given exhaustive talk on the various tidbits regarding the use of laboratory animals and handling of laboratory animals. It was very informative. So my topic is basically the use of laboratory animals in biomedical science. So I'll be starting with this one. As far as the laboratory animal is concerned, we all know that it has been used for a pretty long time. It's not now only. It has been there for quite some time. And we are dependent on these laboratory animals, though for any because mainly most of the research which is done is basically for the target species is human being. And for any things to be done, there's a need to do some preclinical trials in laboratory animals. Although there have been instances where we cannot extrapolate directly the findings from animal to human being. Ultimately, you have to do some clinical trials. But still, the animal experimented is one of the very important aspects of drug development. Not in drug development, any biomedical science. So let us go forward. Now, this particular slide, which shows you, gives you about a little story which I would like to share with you. That is the elixir of sulfanilamide. So this particular, we all know sulfanilamide. Sulfanilamide is basically sulfonamide, which is derived from the azodide, tontosil, and it has a very good antimicrobial property. and it has good especially against streptococcus. And people have used this earlier, but the problem with sulfanilamide is that it is not easily soluble. So people who use this as an antimicrobial agent has to use huge amount of sulfanilamide, and it was very expensive at the time when it was first discovered. One particular company came out with a soluble form of sulfanilamide, and they started using this particular drug combination, and they found that this sulfanilamide was very effective, and the human got treated with from infection from streptococcus. But later on, they have seen that most of the people who were treated with this sulfanilamide, the elixir of sulfanilamide, suffered from your kidney problems. And many of these people died due to kidney failure. Later on, when they found out what solution they've used to make the sulfanilamide soluble, they found that they have used ethylene glycol. And we all know ethylene glycol is highly nephrotoxic. It is converted to glycolic acid, which is highly nephrotoxic. So people have started to realize that directly giving drugs or any other substances to human being pose a threat to the life of the human being. So it was made mandatory that any drug to be used in human being has to undergo a series of experiments before they are allowed to be used in human being. So that was the turning point, and since then people started using animal for animal experimentation. So this is a short story which highlights the importance of animal experimentation. Then next, we already know about the tragedy of thalidomide. So we understand that there are certain drugs which can cross the placental barrier at the developing fetus and cause deformities in growing fetus. So any drug which are supposed to be prescribed for during pregnancy has to undergo some teratogenic toxicity. So it was mandatory since then that any drugs meant for human use or animal use also during pregnancy has to undergo some animal experimentation, mainly for teratogenicity. So that is another milestone which move. Now let us come to the actual amount of animals which are being used for animal experimentation. So we often say that if you see the number of animals which are used for animal experimentation, we see a large amount of mice are being used. After mice is the rats. Then after rats, you'll find amphibians. Dr. Sole has already given about the zebra fish. Yes, now even earlier also you find the fish were used for various kind of animal experimentation. But when it comes to pharmacological studies, it is a recent origin. Only 5 to 6 years earlier we have started using zebra fish, but we are using nowadays. Then after the fish, we have these birds which are used. So this particular slide shows you the number of animals which are being used for animal experimentation. Now we often do research, and why we do this research? Number one, for gene expression. Then we do for genetic cloning. Then we do for transgenic animals. For example, we use laboratory animals because they have a very short life, and their breeding also is very fast. So it is more convenient to use laboratory animals for transgenic animals. Then we also use for stem cell research these laboratory animals. Then we use teratogenicity. I've already discussed about that. Then we shoot cancer research. Yes, you'll find certain animals are very, very convenient for doing cancer research, especially in vivo cancer research. That is, we have mice are usually you'll find they are very susceptible to virally induced tumors. Then we also use animals for diagnostic purposes. We use animals for production of vaccine and sera. We use testing animals for infusion fluid, pharmaceutical products, for example. Then we use animals for toxicological studies. Yes, we have already said the importance of doing the preclinical trials of preclinical animal experimentation. And here mainly we do the toxicological studies. Then another important aspects of animal experimentation is the animals and drug safety testing. So this is mandatory, and we all understand now the importance of doing animal testing before they are either fit for animal use or fit for human use. So they have to undergo a series of animal experimentation. Then only the drugs or the molecules are are given your permission or license for your human trial. So what are the various drugs safety testing that we do as far as the animal experimentation is concerned? Number one, acute toxicity, we all know that. Then subacute toxicity, then chronic toxicity, reproductive toxicity, mutagenicity, carcinogenicity, primary irritation testing, then antigenicity. So these are some of the experiments we often use animals for doing the drug safety testings. Then another aspect which we have where animal are often used of, that is, we all know about the cloning. So this particular experiment, which you all know, that is Dolly the sheep, which was produced by cloning method. So so you see, these all type of animals are being used for animal experimentation. Then we come another to one. This is you people may not recall this particular dog. The name of this dog is Laika. So this dog is the first animal, even before human being, to visit outer space. So this animal was used in the Soviet Union rocket, which was fired to go to outer space. Of course, the animal did not survive. While returning back, the animal died. But yes, this particular animal, Laika, is the first animal to go to outer space, even before human beings have ever ventured out into outer space. Then, so we have said about the importance of animal experimentation. So let us, it is impossible to tell you everything about all the laboratory animals. So we will try to tell you some of the laboratory animals which are being used for animal experimentation, routine animal experimentation. So what are these common laboratory animals and how they can be, which can be easily bred in your institute? So first we have the mouse, musculus. So you have smallest. The importance of this one is that it is smallest. Since it is smallest, it will need very less amount of the drug, because the new drug entity which you get, it will be in very small amount. So as a result, these animals is convenient for such small molecules or small amount of drug which is available for you. Number two, is this easily bred? Yes, one of the advantage of using my mouse is that it is easily bred. You only provide the necessary facilities. You'll find you have a colony of this mouse in your lab. Of course, someone has to be very careful with regards to their breeding and their hygienic and their management. Then, yes, I've already said nothing is prolific. Yes, from one litter size, you'll get 9 to 12 numbers of pups. So this is the advantage of using mouse. Then it is cheap, of course. Then easy to handle. We all know that it's easy to handle already. Dr. Sole has shown you how to handle this one. Then another advantage of using my mouse is that it is a small in size. Since it is small in size, you need small dose of the drug. So this is another advantage as far as the mouse is concerned. Now, what are the various uses of mouse? So as far as the various uses of mouses are concerned, we have already some thing has already been said by Dr. Sole, but let us go and visit the other uses of these animals. Number one, in acute toxicity studies, yes, as far as the conventional method of finding the acute toxicity, that is by either by the your Least-Wilker method or the Garber method or Miller and Tainter method. So we often use mouse as a laboratory animal, but it's not necessary that you can use only mouse. You can also use rat, but it is more convenient to use mouse. Similarly, OECD guidelines also tells you that you can use mouse. So this is an animal which is very convenient to do acute toxicity. The second studies that we often use mouse is the insulin studies. Though nowadays we prefer a bit larger animal to do any anti-diabetic studies or insulin studies. A mouse is also a very good model for doing such studies. Then another test that we often use mouse is the analgesic test. So we have seen there are different kind of analgesic tests. Mainly, for example, your reading syndrome, for example, then the Addis hot plate method for analgesia. So we often use mouse. So mouse is convenient for such kind of studies and analytic studies. Then it is also a very good model for general screening of chemotherapeutic molecules, because it is since the animal is very small, so it requires just less number of your drug. So it is more convenient in that case, because the animal is smaller, so you need less dose since you have very small amount of the drug molecule. So these are advantages. Then, as far as the genetic and cancer studies, also we use mouse, because genetic studies, we have already said, since the lifespan of these animals is very short and the breeding time is very short, so the expression of various phenotypes are also very, very fast. So it's more convenient. At the same time, I've already said about the cancer research. So mice is a very good model, especially for virally induced tumors. So there are various tumors can be generated in mouse, which is much more convenient in this case. Then, since we have said that the breeding is very short, so teratogenicity test also is very convenient in case of mouse. We often do this kind of studies in mouse, but of course, it has been observed that certain drugs which may show teratogenicity in mouse may not show similar teratogenic effect in human being. It gives an idea that these drug may have, or these compound may have teratogenicity effect if they show in mice. Then we have another. So some of the common mouse strains which are available for use is, we have already said, common albino mouse. We all know that is available everywhere. Then we have the BABC mouse. BABC mouse is a mouse which have compromised immune function. So this is a mouse which is where it's used for various type of infection, and also various type of your anti-cancer studies, and also other types of studies. So it is much more convenient because the immune system is compromised. As a result, the mouse can be used for different type of your infectious disease. Then we have the OB mouse. OB mouse is basically a obese mouse, which is mainly used for basically your anti-diabetic studies. In this particular mouse, you'll find the gene for expressing the leptin is mutated. As a result, what happened? The animal is hyperphagic, it keeps on eating, and also is obese. So this picture, which is showing you out in the slide, on the right side is the normal mouse, and on the left side is the obese mouse. So you'll find this almost 20 times much bigger than the normal mouse. It's a diabetic mouse. Remember, it's a diabetic, so it is helpful for anti-diabetic studies. Then we have next NOD mouse. NOD mouse is a non-obese diabetic mouse. So this is a genetically the altered mouse, which is diabetic, but it is not obese. We have one example out here. I'll show you. So you see, this is the NOD mouse, which is not unlike the OB mouse, which is obese. So this mouse is not obese, but it is diabetic. Then we have KK and KKA mouse. So these are also different type of mouse which often use. Then diabetic mouse. This is another mouse which is used for diabetic studies. Then we have New Zealand O mouse. This is another one which is often used for various studies. Then we have the nude mouse, where you'll find this mouse is basically is totally devoid of immune system. It is prone to infection, so it is handy for anti-cancer research. It is also for immune suppression, because this particular animal is devoid of thymus, so the T cells are not available, so they are prone to infection. So the various and this mice is convenient for certain type of anti-cancer research. So we often use these mice for different research work. Then we have another mouse, that is another mice. This is a black mouse, which is C57BL/6 mouse. So this is a mouse which is very often used nowadays. So this mouse is a bit, if you compare with the other mouse, this mouse is much more, it has a bad temperament. It is not docile, but there are certain advantage of using this mouse, because in certain type of this mouse is prone to addiction, morphine addiction. Then it's also it is also addicted to alcohol, for example. So such kind of studies you can do in these animals. At the same time, these animals is also susceptible to your dextran-induced sulfate sodium induce your colitis. So we can simulate the human colitis or inflammatory bowel syndrome in this animal. So in such animal model, this animal is very convenient to do such studies. Then we have so this is basically one of the your IBC cages that we have in our lab, in our animal experimentation room, where we put both the we have separate for mouse and separate for your rat, where we house these, especially the Babsy mice and the nude mouse, in individually ventilated cages. Otherwise, if you do not house this animal in separate or individual cantilever, they will be prone to infection. Then next we have the rat, the Rattus nervicus. So this is a commonest lab animal. We all know that this is a very common animal. So the animal has greater sensitivity as compared to the mice. Then you'll find another advantage of using rat is that we can extract tissues, and we can also do some surgical procedures in mice, which is often not very convenient in case of a mouse, because of smaller in size. But if you compare, the rat is much more larger in size than the mice. So if you want to extract some tissues, or if you want to do any surgical procedures, this animal is convenient. Especially, for example, whenever we do any your bioassay, for example, the colon, if you want to take the ileum of the rat, so you'll find for any bioassay of substances which are having cholinergic activity, so in that case, usually we use the rat your ileum. Similarly, for doing any studies of co-transmission, for example, where we often take the vast difference of rat and give electroshock, and from that we can easily find out two type of contraction in the rat vast difference. So we often use this animal for to extract the different tissues from the animals. Then, as another important factor of using the rat is that since it has a big tail, already Dr. Sole has said about the difference between the Sprague Dolly and Wister rat, but at the same time, it has a bigger tail as compared to the other animals. So it helps in handling the animal, number one. And number two, you'll find it has a tail vein from which you can easily get sufficient amount of blood. So it is convenient to extract blood from these animals through the tail vein. Then, unfortunately, unlike the mouse, which we have already said that mouse can be easily we can easily induce tumor with the help of virus. In case of rat, this is not and the animal is not very sensitive to virally induced tumor. But at the same time, some chemically induced tumor you can easily produce in rat. But virally, you find it's difficult to produce tumor in rat. So that is the disadvantage. Yes. Okay. As far as the uses of rat is concerned, it is much more convenient to use rat for psychopharmacological studies, because the expressions are more easy to understand in case of rat as compared to mouse. So we often use this particular animal for psychopharmacology studies. Then assay of hormones. Yes, this is another animal where for various hormonal assay, the animal, since the size of the animal is larger, so it is better than the mice. Then blood pressure, already Dr. Sole have shown you about the cannulation of the carotid artery and fixing to a manometer. So that is invasive type of blood pressure we often use by whenever we cannulate the carotid artery. But nowadays, of course, we have the non-invasive method where we use the tail vein, the tail to put the tail cuff, and we can easily monitor the blood pressure without doing any invasive techniques. So rat is much more convenient than the other animals when it comes to blood pressure. Then study on gastric acid secretion. Yes, again you'll find the rat is another very good model for gastric acid secretion. That's gastritis or hyperacidity. All you have to do is to deprive the animal for 72 hours from food. Then you'll find the animal suffers from acidity. The animal suffers from gastric acid, increased gastric secretion. But at the same time, if you want to, ligate the pyloric sphincter of the stomach, you'll find there is increased secretion of acid, which may lead to gastritis in this animal. So this animal which is very convenient for gastric acid secretion and similar studies. Then another studies which we often use rat is the analgesic test, for analgesics. Already we have said that the mice is a good animal model for analgesic test, but at the same time, rat can also be used. We often use rat also for both in the Addy's hot plate analysometer, and also this is a very good animal model for anti-inflammatory studies with the help of seismometer. So we can easily induce inflammation with the help of carrageenan in case of rat. So we often use rat for analgesic and anti-inflammatory test. Then again, for acute and chronic toxicity studies, we often use this rat. This is a good animal for this models. Then neonatal studies, yes, for neonatal studies we also use rat, especially since the litter size is good. We can observe the various aspects of the pups during the studies. Then hepatic studies, yes, this particular animal is suitable for hepatotoxicity. We can induce hepatic damage by using various chemicals. Mainly, we often use, of course, carbon tetrachloride is a very good model. Then we can also use your paracetamol along with your endotoxin. So you'll find the animal there is damage to the liver whenever you use this paracetamol, high dose of paracetamol with endotoxin, or we can also use carbon tetrachloride. So this it's a convenient animal for doing your hepatic studies. Then teratogenicity and carcinogenicity. Yes, this as I've already said earlier with regards to mice, we have seen the breeding is very fast. So for these kind of studies, it is much more convenient using these small animals rather than using larger animals. At the same time, the carcinogenicity stuff we can do, especially chemically induced cancer, we can easily produce in rats. Then another use of the rat is the use of isolated tissues. I've already said about that. Especially, for example, we often use the rat ileum, we use the rat vas deferens. So these are some of the tissues we often use for bioassay. Then some of the common strains of rats which are often used for various studies. These are given out here. Vista rat, already my friend Dr. Sole has already said about that. Then Lewis rat, this is a rat which is very good for adenomas. They are more prone to adenomas. You can easily produce them. Then sprout dolly rat, already he has said it is as compared to the Vista rat, sprout dolly is bigger in size, but is much more docile. On the other hand, you'll find the Vista rat is much more convenient for cardiovascular your test. But on the other hand, Sprog dolly are fit for many other type of animal models. Then we have a biobreeding rat. This is a rat which is basically a diabetic rat, which have often been used. Then we have long events rat. It is mainly used for behavioral and obesity studies. Then we have Zucker rat. Zucker rat, again, you'll find it interesting out here. Zucker rat are two types of Zucker rat. One is Zucker obese rat, and one is a Zucker non-obese rat. So Zucker obese rat, which is also known as Fafa rat, it is basically a rat which is obese. It is hyperphagic. It can eat. It eats a lot and grow in size. And the animal reveals hyperlipidemia. So it's a very good study in the animal model for drugs having hyperlipidemic effect, anti-hyperlipidemic effect. Similarly, the Zucker non-obese rat is also a diabetic rat. It is hyperglycemic. Then we have the hairless rats, so Rowett nudes, then fuzzy rats, the Shawn rats. So these are all rats which are basically have compromised immune function. So you can use these animals for various type of your anti-cancer research, and also your various type of infection you can produce in these animals. Then we have another rat, that is RCS, rats inherited retinal degradation. So this is a rat which is very good for any test for retina damage. So they spontaneously, after a certain period of time, you'll find these animals undergo degradation of the retina. So you can use this animal for any studies relating to the retina, degradation of the retina. Then we have another one, shaking rat, Kawasaki, that is shaking of the body and ataxic paralytic rat. So this is another rat where you'll find there is it shows neurological symptoms, which is characterized by shaking of the body and paralytic gait. Then we have the next guinea pig, Caviar porcellus. We have also said. So this animal is often synonymous with animal experimentation. Though if you go back to my previous slide, we have seen that the guinea pigs are hardly used animals nowadays, except for very few studies, as Dr. Sole has already said that. But we hardly use these animals other than for few experiments. But when it comes to animal experimentation, we often say this is a synonymous with animal experimentation. Then it is, as you have seen now, this animal is larger than the rat and the mouse, so it's much more convenient. It is very docile. It is biologically different from the rat. Then easy to keep in captivity. Yes, you'll find many people have kept this particular animal as pet animals in the house. So because it is docile, it is easy to maintain. There's an advantage. Then it has a long gestation period and low litter size. Very docile, I've already said that. Now, users, we have bronchodilator studies. So this animal is highly susceptible to bronchoconstriction. So if you want to study any drug which have anti-asthmatic property or which have the property to dilate the bronchi, so this animal is very convenient because you can easily produce bronchoconstriction in these animals. There's an advantage. Then it's also used for immunological studies. Yes, it has been used earlier, but of course nowadays the use of this particular animal is coming down when it comes to Then another use we often use these animals is the local anesthetic. So the animal is often used to screen molecules or drugs which have a local anesthetic property. So it is much convenient for this animal, because if you give a prick on the animal, you'll find the animal squeaks. So if the drug has any local anesthetic property, you'll find the animal will not, even if you give a prick, the animal will not squeak. That is a certain procedure how to do it, of course. But this is an animal where we often use for screening local anesthetic property. Sorry. Then another one, digitalis. This is again for studying the effect of drug on the heart, mainly the cardiotonic effect of the heart. We often use this particular animal. Then hearing impairment. This is another animal where you find we often use for experiment on hearing impairment. Then another important thing with regards to guinea pig is that the experiment on oxygen consumption. The animal is highly susceptible to deprivation of oxygen. And now, if you hold the animal tightly and squeeze it, and if you are not very careful, you'll find the animal dies in your hand itself. Since the animal is highly susceptible to oxygen deprivation, if you hold it tightly on the thoracic cavity, the animal will not be able to breathe. So, as a result, the animal dies. So, any experiments relating to oxygen consumption, so these animals is a very good model for such kind of studies. Then, it's already said, Dr. Sole have already said earlier, the study on tuberculosis. So, this is a drug susceptible to tuberculosis. You can do the studies. Then isoniazid toxicity. There's a good animal experiment for doing any toxicity of isoniazid. Then isolated tissues, yes, guinea pig, we often use the guinea pig ileum, for example, especially for histamine. So this animal is highly susceptible to histamine. There is not only as far as the bronchi or the respiratory system is concerned, even if you'll find the guinea pig ileum has high amount of histamine receptors. So any compound which have either histaminic effect or anti-histaminic effect. So these particular animal, guinea pig, is very convenient for that. At the same time, the guinea pig tracheal chain, we often use guinea pig tracheal chain for bioassay. So again, here we find out the bronchodilatory effect of the compounds. We can use this particular animal trachea and make some rings with this particular trachea and see the effect of the bronchodilators. Then again, you find, as already Dr. Sole has said earlier, that the animal cannot synthesize vitamin C. Similarly, like human being, the guinea pig also cannot synthesize vitamin C as compared to other animals. So these animals requires vitamin C. So I have seen many laboratory animals where they cannot maintain guinea pig. But when I tell them whether they have given vitamin C or they have given fresh food to the animals, they say no. Then when I prescribe them, then they have been able to maintain a colony of guinea pig. So these animals highly susceptible to guinea pig vitamin C deprivation, since it cannot produce vitamin C like other animals, for example. So you have to give vitamin C to these animals in order to maintain a colony of other or fresh fruits. Then we have another hamster, that is Mesocriticus auratus. So this is an animal which often used now, of course, not very popular, but it is used for certain type of studies. So chunky body and shorter legs. Yes, it's a very chunky body and very small legs, short legs rather. Then a fluffy tail. The tail is highly fluffy. Loose skin and dense fur. Yes, this is another thing. The skin is very loose. It is almost similar to like your rabbit, where the skin is very loose. Here also you'll find the skin is very loose and the fur is very dense. It's relatively prolific, like your other animals, like the rat and the mice. So users are concerned, this very useful in biological studies. So for viral study, this particular animal is often preferred. Then cancer research. Yes, this particular animal, you'll find the hamster is very often used in cancer research. The nutrition also, because you'll find this animal is highly susceptible to any deprivation of your food. So it's often used. The genetic research, it is being used. Then pharmacology, yes, we have often used these animals in pharmacology, especially for anti-cancer research. We often use that. Even for virology, we use antiviral drug. Then toxicological studies, yes, but of course nowadays there's a shift from hamster to other animals, especially rat and mice are more preferred when it comes to toxicological studies. We do not use these hamster nowadays for toxicology. But earlier, yes, we have used especially for subacute and chronic toxicity, hamster were used. Then another area where this particular animal is being used is to reproductive physiology, to study the reproductive physiology of animals. So these animals are being used. Then another, yes, this is another very important aspect of hamster, that you can easily produce diabetes in these animals. So to see the anti-diabetic effect of drug, we often use a hamster, which we can induce the diabetes either chemically or by genetical modification of the hamster, or even also by giving high caloric food. You'll find you can produce type 2 diabetes in hamster. Then immunological studies, these animals are being used. Then another animal which is used for pharmacological studies is the gerbil. It's also known as a desert rat. So Merionus ugulicatus cutelatus, as a small rodent. It's a very small rodent, almost like a mice. Then it's known as a desert rat. It has a crepuscular habit. Crepuscular habit means the animal is either very active in the early morning or in the dawn and the dusk. The animal, rest of the time, you'll find the animal be sleeping. So it is only active at early dawn and at dusk. So those animals which are active during the dawn and the dusk, these are known as crepuscular. So these animal is crepuscular. These animal is fit for your studies on biological clock, that is circadian rhythm. Okay, so it digs tunnel, not aggressive, rarely bites, clean, less odor. One thing is that these animals, you find the desert rat or the gerbil has a good temperament. It is not aggressive. It rarely bites. And another important factor why these animals are also kept as a pet is that they do not emit any odor. Okay, unlike if the other animals, will find mouse, for example, you find there is a distinct mouse odor. So all animals will have some odor, but this particular rat, that is the gerbil, do not have any odor. As a result, these animals preferred, and many people keep these animals as the pet animals. Okay. Then the eyes of these animals are prominent as compared to the other animals. Then as far as users are concerned, gerbil is mainly used for epilepsy studies. So these animals are susceptible to your epilepsy. You can induce your epileptic seizures either by using electroconvulsometer or by using some chemically induced convulsions. So this animal is being used for epileptic studies. Then another study the animal is often used is the anti-cancer research. You can produce cancer in these animals. So this animal is being used for anti-cancer research. Then rabbit, already much have been said about the rabbit. We also have some clear idea with regards to rabbits. That is Arictelagus cuniculus, this particular rabbit. So it's a very docile animal. We know the rabbit is a very docile animal. And then it is also prolific. But one thing is that you'll find the prolific, unlike the other animals where the ovulation is spontaneous. In rabbit, it is not spontaneous, it is inducible. Only during coitus you'll find the female rabbit will lay eggs. Otherwise, it is not spontaneous like other animals. So this animal is also very prolific. Of course, you won't find very big litter size. 2 litter size you'll find very often. Then it has a short gestation period. Unlike the size of the animal, you'll find the gestation period very short. Then another important factor with regards to atropine, with regards to rabbit, is that you'll find these animal certain drugs, anticholinergic drugs like atropine, are not effective due to the presence of an enzyme which can metabolize atropine. So that is atropinase. So if you give the drug by parenteral route, it will not be effective. But on the other hand, if you give by directly into the eye, you'll find then it will show some effect. And this particular resistance to atropine is much more seen in the colored breeds of rabbit, not the white breeds. Only the colored breeds you'll find they produce this particular enzyme which can inactivate atropine. While on the other hand, the white one, the atropine is effective, but not in colored breeds. Then carotid depressor nerve, then you'll find another important factor is that they have the cardio aortic nerve, which is a depressor nerve. So if you stimulate this nerve, you'll find the heart stops. So this is one of the factors which is different from the other animals. No adrenergic vasodilator nerves are seen in this animal. Then as far as the uses is concerned, the primary use of the rabbit is pyrogen testing. Those fluids which are meant for your infusion fluids, which are meant for to be given by parenteral route, especially by intravenous route. So these tests, these fluids should be free from pyrogens. So any fluids which are meant to be given for infusion has to undergo pyrogen testing, and we do this pyrogen testing in rabbits. So this is an ideal animal for doing pyrogen test. Then another studies with capillary permeability test, we often use the rabbit in this case. We find out the permeability of the capillaries in these animals. Then insulin and hypoglycemic studies, yes, we though the rabbit is not actually a very good model, but we often use a rabbit for anti-diabetic test, and it can be used. It's not that it cannot use, but so we also use these animals for your anti-diabetic. Then another test we often use this rabbit is the Curary effect, that is the neuromuscular blocking ability. The ability. So this animal is very susceptible to the blockage of the neuromuscular, that is mainly the nicotine receptor and the skeletal muscles. So we use this particular animal for such kind of Curary effects. Then sex hormone, assay of sex hormones, because this is a large animal, you can get large, more quantity, more volume of blood. So you can do various sex hormone assay in this animal. Then another important area where this particular animal fits in is the dermal toxicity. So we often do the various type of dermal toxicity in rabbit and in guinea pig. So these two animals are very suitable for dermal toxicity, especially for irritation test, hypersensitivity test, dryness test. So all these tests, including the test for irritation of the conjunctiva, we often use these animals, especially rabbit and the guinea pig. So these animals are very suitable for dermal toxicity. Then, as already the Dr. Sole has shown you, we can easily draw blood, especially the marginal ear vein of the rabbit. So it is much convenient to you can easily see the vein, so you can easily puncture it and collect sufficient amount of blood. You can also use some cannula and you can cannulate it so that it is much more convenient as compared to other animals. Then serological works, yes, it has been used earlier. Of course, nowadays the use of animals for serological works has come down, but it was used earlier. But still, some labs are still using these animals for serological works. Then another aspect where we often use is the teratogenicity and embryotoxic agents. So to find out the ability of a compound to produce deformity in the growing fetus, or if it has any effect on the embryo, toxic effect on the embryo. So we use rabbit in this case for such kind of test. Then as I've already said about that, this is a larger animal. So reproductive studies are much more convenient. And at the same time, we all know that these animals is basically does not spontaneously ovulate. So it is induced. It is only it ovules by induction. So this is a difference between the other animals as compared to the rabbit. Then we also use various tissues from the rabbit for bioassay, especially, for example, we use jejunum, the ileum. So various tissues we often use from rabbit, mainly the gastrointestinal tissues. So it is much more convenient, mainly for assaying substances which have adrenergic effect, either adrenergic or anti-adrenergic effect. So we use this rabbit gastrointestinal tract. Then other animals which have been used for various studies are, for example, cat, though it's not a regular laboratory animals, but a cat is a very good animal model for cardiovascular studies. So it has been used and will be used in future also. Uh, their cardiovascular system are more responsive as compared to the other animals. Then dog, yes, dog we have used for various purposes already. Of course, nowadays there's a due to ethical reasons we do not use dog. But earlier, for example, when it comes to blood pressure and other effects, we often use dog. But nowadays, due to ethical reasons, we do not use dog. But yes, there has been instances where dogs are still being used, especially in some cosmetic cosmetic studies, because they also have a very sensitive skin. So for any to irritation studies and hypersensitivity studies, these animals are often used. Dog. Otherwise, we do not have much use of dog as far as the other tests are concerned. Then we have another, that is the amphibians. Yes, we have used amphibians earlier very often, but nowadays we have seen there is a blanket ban on the use of amphibians. Amphibian has been used mainly, for example, a frog we are often used for doing seeing the effects of drug on the heart. We have the strop techniques. We have the various heart perfusion test, the strop test. We use the frog heart. Then we also use for local anesthetic effect. We used the frog legs earlier. We block the sciatic nerve and see the effect of these on acid. Then we often use the rectus abdominis muscle of frog to see the effect on the skeletal muscles. But nowadays, of course, there is a complete ban on the use of amphibians. This is mainly due to the fact the number of amphibians is decreasing at an alarming rate. Earlier, you find whenever the rain starts, you'll be hearing croaking of the frogs everywhere. But nowadays, you hardly hear any frog. In fact, there is a frog is an important biomarker of your environmental degradation. So as the number of frogs has come down, that means the environment is in danger because it cannot sustain this frog, which are a very important part of the wildlife system of our planet. So the number of frogs have come down drastically. So there's a complete ban on use of amphibians for any animal experimentation. But yes, there are certain breeders who have the license to breed and supply you, and you can do those tests from these breeders. At the same time, you can also collect amphibians from the wild, provided you get the permission from the Wildlife Warden of India, who is the nodal officer as far as the use of wild animals or any wild animals, fish, amphibians, or any wild to do any kind of studies. So he's the only person responsible for giving the permission for that. But it is very difficult to get the permission to run that. Then fish, we have already said about fish. Dr. Sole has said early, especially with regards to zebra fish. Yes, zebra fish is is replacing the mice nowadays in various studies, especially studies of the nervous system, and of course, and also in cognitive dysfunction, and also in depression. So the fish has been found to be very useful in studies, especially with regards to neurodegenerative changes, and also the depression studies that are being used nowadays. Then other animals which have been used, of course, is transgenic animals. It is coming in a big way. There are various animal models which have been created by introducing or removing certain genes of these animals, which mimics human disease. So we have transgenic mice where mice have been manipulated, the gene has been manipulated in these animals, and they can produce or mimic certain diseases of human being. Then we have rats, for example. Similarly, we have many rats which are there. Then pigs are also, you'll find transgenic pigs are there. This is an example of a transgenic pig. But the problem with this transgenic pig is that you'll find these animals suffer from arthritis. So this is one of the biggest problem which have been encountered, which have been seen whenever you use transgenic pig. They are susceptible to osteoarthritis. Remember. Then cattle also, there are your transgenic cattle. Then fish, yes, fish, especially you'll find as far as India is concerned, we have been able to produce a transgenic fish mainly by CIFI, Central Institute of Fishery Research in Mumbai. So I have been able to come out with a transgenic fish. So these are some of the animals which are often used nowadays in various type of pharmacological studies and for biomedical studies. So these are some of the parameters, normal parameters with regards to the various laboratory animals which are given out here. You can go through it. It's given out there. So any deviation from this means that the animal is not good. So here you'll find the various physiological parameters: rectal temperature, heart rate, your systolic blood pressure, then diastolic blood pressure, the blood volume per ml, then respiratory rate, tidal volume. So these are all mentioned here. You can go through it, and this will become very handy later on when you work with animals. With this, I think I come to the end of this particular talk. It would have been better if we had face-to-face contact, because sitting in front of a computer and saying a lot of things is not very effective. But still, we have gone through a very difficult time, and this is the only way we can communicate nowadays with each other. So I thank all of you for giving me a patient hearing. And if you have any doubts, you can please clarify. You can ask now.
Thank you. Thank you very much, sir, for a very elaborate presentation of various conditions and in what species those conditions can be studied. You have explained in very much details, and I think participants must have got benefited, and those who are planning their studies, so they will be having getting a idea of the best choice lab animal for their studies from this. Sir, we have a question in the chat box. So this was the first question from Ms. Shweta, that why it is easier to produce a diabetes in hamsters as
Well,
compared.
they are more susceptible. The pancreatic cells are more susceptible to your destruction by various chemicals. You find, especially you find when you give certain chemicals like alloxane or streptozotocin. So they destroy the beta cells mainly by producing some free radicals. So these beta cells are more susceptible to these chemicals.
Yes, sir. And one more question was there by Priyanka, that which organ system in the mice and rat resembles the human beings?
Mice and rats, organ
Yes, sir.
system,
Yes.
human being.
Yes.
You cannot exactly say which resembles human being, because as I've already said, the various systems I've already said, because you cannot extrapolate findings from lab animals to human being. Okay, so though there is some similarity with regards to various systems. For example, we have the gastrointestinal system, which is much more similar to human being, unlike the other animals. So this is for various studies of gastrointestinal tract. So this is an animal which is much suitable, especially rat. I have already said for gastric acid secretion. So rat is a very good animal model for that. So other systems are not similar to human being. So some of them, but at the same time, rat you'll find it doesn't have this particular gallbladder. Okay. So you cannot directly simulate and say that it is the systems are similar. So these are two different species totally. You cannot directly correlate the various system. Okay.
Thank you, sir. Thank you for your nice lecture and very nice presentation.
Thank you.
Thank you, sir. Thank you, sir.
Yes, sir. We
Nice
thank
to
you
see you.
for giving your time, valuable time, and guiding our participants. Now we have the next speaker for today's. That is Dr. Pravin Bankar, who happens to be from this same institute. And I would request Dr. Hazare sir again to introduce the coming speaker. Hazare sir, please.
Dr. Pravin Bankar is working presently as assistant professor at Department of Animal Genetics and Breeding at Postgraduate Institute of Veterinary and Animal Sciences, Akola. He did his MVSc and PhD in Animal Genetics and Breeding from this institute. He has got 12 years of experience as assistant professor. He has guided one PhD student, and as a minor guide, he has guided more than 10 students. He has got 10 research articles in the national and international journal of repute. He has published 5 review articles, 12 technical articles, and several abstracts. He is a reviewer of several journals, mainly Indian Journal of Animal Sciences, Indian Journal of Animal Research, etc. He is a member of several professional societies like MSVC, then AAIVR. He has organized several trainings and seminars. He has got 2 Best Paper Awards and 2 Best Poster Presentation Awards in national conferences. He has got Dr. B.R. Ambedkar Personality Development Award in 2006. So, Dr. Pravin Bankar.
I would request Dr. Pravin Bankar to kindly start his presentation.
Am I audible?
Yes.
Shinde sir?
Yes, you are audible.
Yes, sir. Okay. Thank you, Dr. Deshpande sir and Dr. Hazare sir, for giving me this opportunity to interact with so many student friends on this online platform of National Training Program. A very good evening to all the participants, respected faculty members who have joined over here. I am Dr. Pravin Bankar. Today, I am here to discuss, to interact with you all on this online platform with a very different kind of subject pertaining to the laboratory animal, and that is, what are the genetic aspects of breeding management in these laboratory animals. I know that many of you, as a graduate, postgraduate student or researcher, are concerned with the laboratory animals. Many of you have directly or indirectly involved in the handling, in the managing the breeding colonies of laboratory animals in various institutes. So, as a man from animal genetics and breeding, I felt some concept of genetics about the breeding management. I should interact with the participants so that whenever they are working with these laboratory animals, they will be smarter enough in their research, in their daily working also. So in that concern, today I am going to discuss with the genetic aspect of breeding management. So friends, we are going to interact, we are going to understand in this session something about the inbred strains, then genetic concept of this breeding and the breeding management. I know that you all are present over here since last, I think 2 or 2.5 hours on this online platform. And it becomes very hectic or very burdensome to me to convey you about the breeding concept, about the ideas of animal genetics which are concerned with this field, but I will try at my level. As earlier faculty members have dictated about what are exactly the laboratory animals, any vertebrate animals which are produced or used in the research, testing, or teaching, and which are genetically defined. So the main aspect is produce or use. So whenever you are concerned with the production of laboratory animals, so you need to have understand various aspects of breeding of these laboratory animals. As earlier speaker also have mentioned, how the laboratory mice and rat are the most preferred animals for testing as compared to other guinea pig, rabbit, amphibians, or non-human primates, or any other farm or carnivore, farm animal, birds. So I suppose that you might have heard about the transgenic mice or knockout mice. However, I am going to discuss with you about the breeding concept, breeding aspect of these laboratory animals. I hope you have seen these common laboratory animals, right from the mouse, rat, monkey, rabbit, hamster, and guinea pig. So the chromosome number, if you go through it, so mouse have 40, rat, monkey have 42 chromosome numbers, rabbit and hamster have 44, and the guinea pig have 64 chromosome numbers. So let us try to understand the concept one by one. What exactly is the strain? When we use the word strain, it means that these are the inbred. Inbred means if suppose there are two dam and sire or mother and father, A and B, the progeny produced from A and B, we can say the animal, these are the brother or sister mating, or we can say sibling mating. So when in this laboratory animal, when we are supposed to mate these siblings for more than 20 generations, then we used to call it as isogenic or genetically identical. Now you know that when in every generation, when the inbreeding is going on, there is a enhancement, increasement in the inbreeding coefficient. So in the first generation, in the third generation, likewise in a 20th generation, we are supposed to achieve about 98 to 99% of the phenotypic and genotypic similarity with the ancestor. So that's why we used to prefer this inbred strain because of their isogenicity, because of their homozygosity. We used to see these inbred strain as phenotypically very uniform. They have a long-term stability. So with these properties, we are concerned with so many derived inbred strain. So we'll see one by one what are that derived inbred strain. So there are several strain like inbred strain like F1 hybrid, co-isogenic, congenic, segregated inbred, recombinant inbred, and consomy. Let us see one by one. Whenever we are saying the F1 hybrid, it means that it is the cross of two inbred strain, two different inbred strain. When, for example, in this picture you can see the homozygote dominant when crossed with a recessive we are getting the heterozygote. Capital A, small a, we can say it as a heterozygote animal. So likewise, whenever we are crossing the two different inbred strain, we are favoring the heterozygosity. And we know that these heterozygote animal, they are quite having a higher vigor. So F1 hybrids are nothing but the product of two inbred strain. Then what exactly are the isogenic? So isogenic are nothing but the homozygous, which are homozygous at nearly all locus, all loci. Say for example, there are 100 of genes are present, and if suppose two animals are having similar or nearly all the for all the locus, they are homozygous. So then in such case, we used to call that two individual, or preferably we can say two strains, as isogenic in nature. However, we may come across with such a two strain which are identical or isogenic for all the loci except one or two loci. So they are different from the one or two locus. Say for example, there are the two strains of the same species, but difference in the coat color. So coat color, we can say it as a one of the trait where the two strains are different from each other. Otherwise, they are isogenic for all the locus. So these are known as co-isogenic in nature. Then what are the congenic? Congenic means when we found the animals in the population which are mutant bearing, means they are having the combination of this mutant A and plus likewise. So the repeated crossing of these mutant bearing animals to the animals of the inbred strain, they are going to impart us congenic strain. So regarding mutation, we used to think about it. What exactly it is? Mutation is nothing but the sudden change, abrupt change or modification in the DNA. It may be either due to deletion or insertion, whatever it may be. But how this mutation is going to hamper or produce the congenic strains? Because even if this mutation is occurring, but the frequency of mutation is quite low. Means for per germ cell, it is to be said that 10 raised to power minus 8 up to the 10 raised to power minus 5. This is very, very low frequency of mutation. And usually whenever the mutation is occurring, it is affecting to the non-coding part of the DNA sequence. So no need to worry about, means as a researcher, you should not bother about that the animals in our population, they are having some mutant alleles. Most of the mutations, these are recessive in nature and are detected much later in their homozygous individuals. And whatever the lethal mutations are there, they are not supposed to transmit to the next generation. And some of the mutations are important and they are used or applied to create some pathological models. So this was something about the mutation. The next inbred strain is a segregating strain. When the strain is having particular allele or mutation which is maintained in a heterozygous state, then it is called as segregating inbred strain. So these are segregating inbred strain. These are maintained by inbreeding, means just sibling mating with forced heterozygosity. Then the recombinant inbred strain or RI strain. So these are produced by crossing two different inbred strain to produce the F1 hybrid, and then following this brother sister mating more than 20 generations. So generally this RI strain or recombinant inbred strain, these are very much useful for determining a specific gene which is responsible for any phenotypic characteristics. Then similar to RI strain, the RC strain or recombinant congenic strain is there. RC strain, here what we are doing the cross. This is also the cross of two inbred strain, but here we are offering the back cross of spring to their parental strain for 2 generations. And then for next 14 generation, we are following the brother sister mating. So these are similar to the RI strain. These are also useful to find out the gene which are responsible for the phenotypic characteristic. Then consomic, or which we can say alternatively it has a chromosome substitution strain. Chromosome substitution strain means this is developed by back crossing a whole chromosome. Here in the third picture, you can see the chromosome number 3 is being used. So what we are doing, we are back crossing. Back crossing means the crossing of progeny with their parent. It is known as back cross in genetic. So back crossing the whole genome. So it is the when the donor inbred strain into recipient background strain. So these are also used for understanding studying the genetics of some complex traits. So friends, you might think about the various strain. Many of you have seen various strains and substrains also, and you may differentiate on basis of their coat color also. Say for example, I have enlisted here certain mouse strain like agouti or brown agouti or black non-agouti, depending on right from the albino, piebald. So here different strain have been nomenclated, have been named with different patterns, and how to remember or how to understand these abbreviations or these nomenclature, that we'll see. Regarding the nomenclature of this laboratory animal strains, you may be reading or going through any literature. You may come across with such nomenclature: CBA/CA, TG/VB8. So very superficially, it seems very difficult to remember, but these are the names of the strain which are self-explanatory. So let us see what exactly these are indicating. So for example, here in this name CBA/CA, this is the name of strain. PG, it is indicative of the strain is transgenic. VB8, it is a symbol, and the sign plus or minus, this is indicating of the genotype. So likewise, we are supposed to nomenclate the strains to get the information in a very precise manner. Say for example, if you have hybrids, how we will identify what kind of inbred strain is there by reading this name of the strains. So for example, here C57BL6J. So this is the cross, this is a maternal strain which is crossed with the paternal strain DBA2J. So here we can identify the hybrid strain which are being indicated with a cross sign. So this is a hybrid strain. Even if you have information about the offspring from the crosses between the F1 hybrid, so you can write down in bracket the whole name, and in front of that F1. F1 is indicative of F1 hybrid. So likewise, we can identify the strain as a hybrid strain when it is having the cross mark. Similarly, these are the names of the strain. We may concise this name by their abbreviation. So here recombinant inbred we can indicate like C into B. C is a parental strain and B is another parental strain. For example, for outbred stock, the holder of the stock which is followed by the name of holder stock, abbreviation of the holder of stock is followed by colon mark, which is indicative of outbred stock, and then name of stock. Likewise, if the stock is carrying mutant gene, then it may be indicated by new, NU/NU. So for inbred strain, this was colon mark was for outbred stock. For inbred strain, we may use inbred strain followed by the sign in bracket, the word F. F, it is indicative of inbred. And if it is F20, so it is indication of the animals or the strains are belonging to the 20th generation. So these are just a symbolic way of expression or nomenclating the strain. Then substrain, if there are substrain, these are simply indicated by hash sign or slash mark. If there are sublines, then these sublines may be means for inbred parental line, a parental strain, and another strain. They may be separated by what kind of this subline are produced. Say for example, here small f letter is indicating of fostered. So it means that the inbred parental strain has fostered the another strain. It may be E for egg transfer, H for hand rearing, O for ovary transplantation, P for preservation by freezing, likewise. Similarly, friends, the co-isogenic strain. Someone has asked about PowerPoint presentation. Don't worry, I am going to share this presentation with this group and with the organizer. So don't bother about it. For co-isogenic strain, the background strain and the co-isogenic strain, these are indicated by a dash mark. And congenic strain, these are indicated with a full stop mark. So the name of strain, they may be separated by a full stop. So we can identify it as a congenic strain. If the name of gene is there or allele is there, it is generally written in italic space. For transgenic strain, it is a very self-explanatory nomenclature pattern. Say for example, in this strain, the first segment is indicative of a strain which is carrying this transgenic gene. The TG, letter TG are indicating of transgenic. Then in bracket, the information, it is indicative of location of the gene, source of the gene, name of the gene, and followed by where the experiment was conducted and number of transgenes were, number of point of insertion where the transgene have performed. So in this way, we can identify the strain on the basis of their nomenclature. Coming to our main objective about understanding of breeding systems for these laboratory animals. So friend, what is the purpose of understanding the breeding systems? For any breeding system, any breeding plan, as a breeder, one has an objective to preserve, to control the genetic causes of variability of trait of interest. Whenever we are performing any experiment with respect to laboratory animal, we are generally applying or we are generally looking towards some traits of our interest. It may be qualitative in nature, it may be quantitative in nature, and we used to observe certain variation in that traits. So that variation is because of their genetic makeup also and some environmental factor also. So that's why as a breeder, it should be our aim to identify or to preserve or to control what are the genetic reasons, genetic causes for this variation. So in case of, it is said that, say for example, in a random mating population, if there is a no selection, no mutation, no migration, we know that the selection, mutation, migration These are nothing but the driving forces which are changing the gene frequency and genotypic frequency of the population. Those who are recalling the Hardy-Weinberg law of equilibrium, they can recall the law statement that in a large random mating population and in absence of selection, mutation, migration, and genetic drift also. So the gene and genotypic frequency of this large random mating population always remains constant from generation to generation. So in a large random mating population, generally or ideally we can say that the mean and variances are constant. However, here we are supposed to apply as a breeder, we are supposed to follow some inbreeding, sometimes outbreeding, sometimes selective breeding. And then if we are applying our interruption, then what are the effects on the genetic variability are observed or supposed to be understood. Here we know that when we apply a selection on the population, whatever is the mean of original population, that is getting deviated because of our kind of selection. We know that when we are selecting the population or the animals from which are indicating their mean values from the population mean, nearer to population mean, we are favoring the stabilizing kind of selection. When we are supposed to select a particular group of animals which are having higher mean value, then it may be a directional kind of selection. Or when, if we consider the normal bell-shaped curve, normal distribution curve, and if suppose we are selecting the individual from the two ends of this normal distribution curve, we are going to observe disruptive kind of selection. So these changes we are generally observing. So here I would like to share something about this genetic gain concept, because many of the students, they are concerned with this breeding management of the laboratory animals, and as a breeder or as a researcher, you should be having idea about this genetic gain concept. So friends, suppose you have a population, we can say that population of laboratory animals at your lab animal house as a parental population. So amongst that parental population, say for example that parental population have some mean value. Say for example, you can consider any trait like birth weight. So whatever is the birth weight recorded for every animal birth, lab animal birth at your laboratory, it may be indicative of certain digit or certain figure, which we can say it as a mean value of your parental population. And then amongst this available stock, if suppose you are going to select those individuals which are having their young one as a higher birth weight. So here we can say that out of 100, say for example 100 animals, you are selecting 10 animals, such animals whose young one are having higher birth weight. So from the original population, you are selecting certain another population with higher mean. So automatically their mean value will be higher than the population mean, isn't it? So here we can say that your selection is making some difference in the mean value of the selected population. So we can say it as this difference as a selection differential, because the changes in the mean value of the population are observed because of your selection. How do you selected your individual, or on what criteria you selected that particular population? And we know that this selection differential it is directly proportional to the response to selection. Because whenever we are considering the heritability of any trait, by multiplying the heritability with the selection differential, we are supposed to receive the response to selection. And this selection differential, it is difference between the mean of progeny of the selected parent with the mean of population. But when this progeny is giving birth to the next generation, and the next generation is giving certain higher ranges of the mean value, then this difference, the P1 and P2, this is known to be the response to selection. And we know that whenever we are supposed to learn the concept of genetic gain, the selection intensity, accuracy, and standard deviation, these are directly proportional to genetic gain. However, the generation interval, it is inversely proportional to the genetic gain. So, how do you select the best of the best animal? It is going to add in the selection differential, or indirectly it is enhancing the response to selection, or in simple terms, we can say you are enhancing the genetic gain. So these were something about the concept of genetic gain. Now I would like to share you about what are the possible effects of inbreeding. Inbreeding, we have already seen when we are following the sibling mating for number of generation, we come to know that the homozygosity is increasing. However, from the original population, we are, with the help of inbreeding, what we are doing? We are making the two subpopulations, population A and population B. So whenever the such subpopulation are forming, then at that time, as per the Bulmer effect, founder effect, we know that there is a loss of genetic variation. So when a new population is established with a very small number of individual from a larger population, then the genetic variation is declining. In case of outcross, what happens? Outcross are the mating between the population, two different population. We are mating the individuals from two different population. So definitely outcross is changing the mean value and increasing the genetic variances. However, the selective breeding means when you are supposed to mate like with like. So here, when you are mating the similar animals selectively, then you are changing the mean as per direction of your selection. And this is going to decrease the genetic variance. However, it is not completely eliminating the genetic variance. Then this is what I am going to share with you all, that what are the possibilities of what kind of breeding system we are going to adopt, and what are the possible effects that are being supposed to observe in the next generations. So when we use unlike animals, or we can say selected mate, two different population A and B, then we used to find the large genetic variance. So here we can say that the combination of the inbreeding and the proper selection system, this will give you as a geneticist a wide variety of methods of controlling the inherited characteristic in this research animal. So what kind of breeding systems are there? So when we say mating of like homozygotes, these are known as incrosses. So we know that if we mate the like homozygote, we'll get 100% homozygotes. The progeny will be exactly genetically similar to their parents. However, if we mate unlike homozygotes, these are known as crosses. So here, the two individual white and gray color, these are different between, they are deviating from for certain trait or for certain allele, then automatically their progeny is having the combination of both parents. So these, unlike homozygote mating, these are known as crosses. Then mating of homozygotes and heterozygotes, these are known as back crosses. You may cross the any combination. So these are referred as a back crosses. Now in case of this third category, when we are crossing the, this homozygous is getting crossed with the heterozygotes. So there is a possibility of 50% homozygotes and 50% heterozygotes. When you are supposed to mate the heterozygotes, then these are known as intercrosses. And we know that as per as the Mendelian principle, we receive the progeny in 1:2:1 proportion. That means we will receive 25% homozygotes dominant, 50% heterozygotes, and 25% homozygotes recessive. So this was about the breeding systems one should know. But here, when we are thinking about the what kind of breeding system we have to follow for breeding our mice or breeding our laboratory animals, here you will notice when the frequency of heterozygotes or probability of heterozygotes decreases, the probability of incrosses increases. Or in other way, when you are following the crosses or back crosses or intercrosses, automatically you are declining the frequency of heterozygotes. Friends, there are two kinds of breeding systems. One is a random mating and another is a hybrid kind of mating, which is naturally occurring. Random mating means in a population, there is equal chance to every individual to mate with each other. So there is no human interruption, there is no human control. This is the what we can say natural mating. So every individual has equal chance. However, in case of hybrid mating, so there are two different populations. So say for example here, in first population, there are white colored mice, and in second population, brown colored mice are there. So these are completely two different sort of populations. And when any individual of either sex, means from male of this first population and female of second population or vice versa, when such crossing occurred, then it is known as hybrid mating. So whenever we are going to or we are adopting any breeding system, we are considering particular traits for our study, and these traits, these may show the variation in two fashion. Say for example, if you are considering the traits like litter size, sex, coat color, we know that some qualitative traits, so they are indicating the discrete kind of variation. You can discretely say sex, male or female, coat color, white. You can classify into some grade or classes: white, albino, or piebald, or Himalayan pattern, or black, or gray. Likewise, so you are able to classify the classes. You are able to grade the classes of that particular trait. Then the variation, it is, we can say this such variation of these traits as a discrete variation. However, there are certain traits like body weight, body size, or lifespan. So these are certain traits which are generally called as a quantitative traits. These are quantitative traits because we can measure that trait in a metric system, and the variation exhibited by such trait, they are known as continuous kind of variation. So for any traits you consider under your research or study, it will show either kind of variation, either discrete or continuous. Now, for this variation, as a researcher, it is your goal as a breeder to find out what are the reason of that variation. If you are providing all of this sort of what we can say managemental condition equally to all the individual, still you will find there is a variation with respect to particular trait in your observation. So these reasons for this variation one has to identify, and these are concerned with the two main aspects. First is a genetic background, another is a non-genetic background. Naturally, whatever is the genetic capacity, genetic genotype of that zygote or that individual, it will exhibit according to that. So that is the background of their genetic makeup. However, certain factors like the changes in the season or feed, whatever you are giving or feed consumed, or exposure to certain external factor like light, noise, humidity. So these are the non-genetic factor. So here we can understand whatever traits which we are supposed to study, these are severely affected with two driving forces or two reasons. One is genetic makeup, another is certain non-genetic factors. And that's why with this knowledge, one has to understand why there is need to maintain the breeding colonies, isn't it? So one has to, or we as a researcher has to maintain the breeding colony to produce the newer lines, newer strain, or to expand the existing colony. This is basic what we can say purpose. Or even you may have to maintain a particular line, or for research one has to supply the animals, and for that one has a stock of such laboratory animals. So for that they have to maintain the breeding colonies to provide the animal for research, for experimental use, or even as a replacement breeder, one has to provide the breeding colonies. Now, as a breeder, what one should know, certain basic things you should know. First is what factors which are required to set up breeding colonies. What are the criteria? So means before establishing a breeding colony, you will have to think about what are the number of animals required at your end, and what kind of sex, either male or female. What is the level of your production? What is the duration of production? Whether you are going to produce or breed the animals throughout the year or for certain period of the year or in particular season. What kind of genotype do you have? And how you are going to maintain the replacement rate. So these are certain factors which are to be considered while setting the breeding colonies. Then in managemental lecture, you will come to know about what are the general breeding information, age of sexual maturity or estrus cycle, estrus period, gestation period, reproductive life. This thing will be covered in by another speaker. Then some characteristic like good and poor breeder female, selection of foster mother. These are the managemental aspect, but one should know about it. Again, you should be very aware about some reproduction performance of some common strain. Say for example, for outbreed strain CD1 or FVBN. So these are the very excellent with respect to litter size. They are producing 7+ progenies at a time. Then the choice for many researchers is C57 BL6. So this strain is imparting 5 to 6 liters in one calving, and there are certain other strain which are very poor. So you should also think about the reproduction performance of common strain. Then another managemental issue about minimum space requirement and cage density. So as a breeder, we should think about this. So with respect to mating cages or mating pattern, generally 3 patterns are being followed. One is a monogamous pairing where one female is mated with one male. So in a cage, the breeder is keeping one female and one male. Another pattern is trio mating or trio pairing, where 2 females and 1 male are kept in one cage. And another third pattern is a harem mating, where 3 to 4 female are kept along with 1 male. Then in such situation, we do think about what is the best scheme to have desired genotype, what is the best the genotyping method, how we necessarily control our mice population, and how do we follow the institution's guidelines of animal care and use committee. So basic principles for managing the small mouse colonies. These are the principles which are provided by reputed laboratories which are working in this area. So we should follow certain principles. Just say, for example, we should maintain a minimum 6 breeding pairs which are representing 2 different generations in our colonies. Then at least 2 generations of a strain should be retained, and we should not eliminate one until next is being produced. Then as a range of breeders, we should keep the 2 to 8 month old mice, because the older mice, these younger mice should be kept, because we may face certain difficulty when we have a older mice for breeding. Then the breeding, monitoring the breeding performance very keenly, backcrossing our strain after 10 generations. Usually after 10 inbreeding generation, you should maintain the substrain divergence by backcrossing. Then certain to avoid the losses due to decline or seizing or catastrophic events. So one should have a contemplate cryopreservation of specific strains. There are certain factors which are affecting the breeding performance that one should understand. Birth defect in a pup. So different strain have varying tendency to have a birth defect. Say for example, in case of C57BL6J, the pup generally reports or generally hydrocephaly condition is observed in that strain. Or another strain AJ, it is having or it is producing the pups with more cleft palate condition. So certain this kind of strain variation that one should understand. Hybrid vigor. Hybrid mice tend to have more or larger liters and than the inbred strain. Then strain specific behavior. Particular strain have their own sort of behavior. Means not all these strains are similar with respect to their mothering ability or mothering instinct. Certain strains are very good foster, and certain strains are very poor in their first litter. So that we should understand. Then mutation and transient effect. Some mutations are embryonically lethal. They may cause infertility, or they may reduce the fertility, or any dysfunction with respect to mammary gland. So these are the very minute points as a breeder one should know. Right from the managemental condition like temperature and humidity. The optimum temperature, best temperature is supposed to be 16 to 26 degrees Celsius, and humidity between 40 to 60%. So that we have to maintain for having a optimum breeding performance of the animals. Then likewise light intensity, light cycle, that you will have to maintain. Noise and vibrations, these are many times these are causing very disturbances and hampering the breeding performance. And this frequently the disturbances due to noise or vibration, it may inculcate the habit of cannibalizing in the mothers. Then barometric pressure, odor, handling, nutrition, feed replacement, health. These are certain, we can say non-genetic factor, but these are affecting the breeding performance in laboratory animals. So how do we improve the breeding performance? We are supposed to replace the breeder at the right time. One should maintain the breeder pair with various ages of replacing and percentage of them by monthly or weekly. Then replacing the non-productive breeders, those animals which are not productive, you should notice them and remove from the colony. Early mating of the mice, when you are supposed to mate the mice, generally when they are 6 to 8 weeks of this age group, it is favorable. Younger mice will breed better than the older one. Then use of the experienced male, many time pairing of young female with the older male, it will improve the breeding performance. Accuracy of breeding record, the most important thing. Again, certain miscellaneous precautions you have to take about the litter fostering, some diet supplementation, mating numerous females simultaneously, even the pregnancy detection, although it is not that much easier task, but you can observe the copulatory plug in the vagina of female after the mating, and on that basis, by expertise, you can understand the pregnancy detection. So record keeping, I will like to mention here, when you are using some pedigree card or card file or pedigree chart, you should maintain the details about the sequence number of that particular mice or that population or strain, sex, phenotype, fate or destiny of that animal, whether it was operated or whether it was culled from the population, dam and information about the dam and sire, strain and genotype, and information about generation, sublines, number of animal born in a litter size, date of birth. So these are very minor points, but you should you are supposed to mention, you are supposed to record. So here, friends, I would like to conclude my this talk with the conclusion that as an experimenter, we may manipulate the genetic variability in laboratory animal or more specifically in mice by means of applying proper inbreeding systems. And when we are following the sibling mating, that is brother sister inbreeding, it has been useful in producing genetically a very uniform population and which yield a large number of inbred strains. The strain may cross to analyze the genetic basis of discrete or continuous traits, and the heart of any colony management program is appropriate breeding scheme. So I would like to thank the organizer who gave me this opportunity to interact with you all. These are certain references which I referred here. And if you have any queries or your kind feedback, so I am mentioning here my email and mobile number. So organizer, if you have any problems, if you have any queries, I think it already, this has been very hectic since after more than 2.5 hours. So no doubt, no problem, dear. If you have any problems, queries, you may also ask in our WhatsApp group. I am there also, and I will try to answer your queries. Actually, the chat box is blank, and I think there are no queries from the audience side. Okay, friends, I would like to take leave from this online platform. I would like to invite Dr. Hazare sir for the
Thank you, Bankar sir, for very elaborative lecture on genetic aspects of breeding and management. Many institutes have the laboratory animal house. So this lecture is very important for those people those who are involved in the breeding. We use laboratory animals, but how they come, what is inbreeding, what is outbreeding, and how parents' line are maintained, and how these animals are bred and maintained. So this is very elaborative lecture given by Dr. Pravin Bankar. I am very much thankful to Bankar sir. I am also thankful to today's guest speaker, Dr. Sushant Sole and Dr. Pritam Mohan. So I declare today's session is over. Tomorrow there will be 3 more lectures from 3 p.m. to 6 p.m. So with this, we are concluding today's session. Thank you. Thank you. Hello, Pritam sir. How are you?
Shall we close, sir? Hazare sir, shall we close the session?
Yes, sir.
Okay, yeah. Thank you.